and working on some aspect of rice genomics. So the title of today's topic is rice genomics and bioinformatics for disease resistance. As Dr. Haleem said that 90%, more than 90% audience will be students. So I tried my best to prepare my presentation in such a way so that a student should be benefited, right? Of course, there are several things, faculties and researchers, and then scientists. They may be knowing very well, but it will be little easy type. I will try my best. So rice genomics. This starts from the rice genome sequencing. 15 years back, we have completed the rice genome sequencing. This is the rice chromosome. India participated on chromosome 11 of sequencing, long arm of chromosome 11. So whatever sequencing strategy we has adopted, the same strategy was adopted for the other countries because 10 countries participated in rice genome sequencing, and the strategy for the whole genome sequencing was same. This is the chromosome, long arm of chromosome 11, and these are the clones which we have prepared. Every country has prepared the genomic library. One laboratory has prepared the genomic library. That genomic library was distributed to all other countries, including India, and we have received those clones which belong to long arm of chromosome 11. So these are the clones 1, 2, 3, 4, 5, 6, 70, 71, 550, 100, more than that. We have received, and these are the name of the clones, and these are the position of the clones onto the chromosome. So our responsibility was to identify the clone of our region, and then after identification of those clones, we did the sequencing of each clone one by one, right? And then we saw that they each clone having some overlap with the other clone. All south clone and north clone are having overlap with each other, and by that way we have announced the sequence. Once the sequence is ready, then what information we have got? These are the sequences which is available on the screen. That the Sanger's method of sequencing we have followed, and after sequencer give the sequence, these sequences are visible in this way, in the color mode also, and in the ATGC also, ATGC mode also. These are the number of nucleotide 210 to 20 to 30, and these are the nucleotides, and every nucleotide has different colors, so that they are double check that these sequences are correct. So from the whole genome sequencing, we have got these informations. Of course, we were knowing that rice is having total of 12 chromosomes, and the size of the rice genome was 390 megabase in size. This was completed by International Rice Genome Sequencing Project, IRGSP, and then they have reported a total of 37,500 protein coding sequences. That is 37,500 genes available on the rice genome. And they have also reported that these gene density was at 9.9, every 9.9 kb, there was one gene. Out of this 37,500 genes, only 61% showed homology with the rice EST. So far means the time which this was reported, only 61% shows that with the rice EST. It doesn't mean that the other are not gene. Of course, by that time, whatever EST database was there, according to EST database, it was matching with 61%. And among these 37,500 genes, 2,800 were rice and cereal specific genes, right? So now the question is arising: what is the function of these genes? 37,500. Why it is not going? The slides are not going up.
Sir, your co-host and your
How to do that?
people and
It is not working. These are the technical problems sometimes we face. None of my board is working. What to do that? Do you have any idea? I'm not very much expert in this.
sir, you try to rejoin if it is a problem.
But I have to come out. No? Should I leave and then can join again?
Yeah, you stop sharing and you start again.
Okay. Pause. Stop.
Sir, start your screen sharing, sir. Yeah, it is coming now. Yeah, it has come.
Again, I'm facing same problem.
सर आप मैक्सिमाइज मत करिए सर, स्लाइड्स को मिनिमाइज कर दीजिए,
Okay.
शायद प्रॉब्लम नहीं होगा.
Is it okay now?
Yeah, it is moving now.
Actually, so many people are joining and they are asking me to admit. So can you stop that? You admit from their side, that
Yeah,
your side.
they themselves will get admitted, sir.
Okay. Okay. Is it okay now? This slide.
Yeah, yeah.
Okay. So genomics is the study of the structure and function of the whole genome. So कहने को तो बहुत छोटी सी चीज है, study of structure and function of whole genome. So what is whole genome? A genome is the study of sum total of all an individual organism's gene. All genes present on the one's individual, that is whole genome. That is all chromosomes, whether it is a rice genome, 12 chromosomes, or human genome, 23 pairs of chromosome. Whatever number of genes are present in the whole genome and their study and their circumstances in which they are being expressed, that is whole genome. And then genomics is the study of the genes of all of the cell or tissue at the DNA level, that is whole genome sequencing, RNA level, that is transcriptomics, or protein level, that is proteomics. Right? All genes are not being expressed. If you will have isolate the DNA and do the PCR with your gene specific primer, you will get the PCR product, but you may not get the RT-PCR product because that particular time gene is may not be present. So we have to find out that at what circumstances our gene is being expressed. The study of all these aspects is called genomics. Why we are going for a genomic study? That is understanding the structure, relationship, and evolution of the genome, isolation of genes and finding their structure. Of course, once you isolate the gene and then see that what are the structure of this gene is having and how this structure is matching with the other genes. Based upon that, you identify and predict the function. Once the function is predicted, you go for the experimental analysis to find out, to confirm whether the predicted function is the same or not. Then understanding an interaction of genes, because there are some genes which is not expressed if other genes are not present in their neighbor, in their vicinity. So how these genes are interacting with each other, how these genes are present, how these genes are activating or deactivating the function of other genes present in the vicinity. All this information we can get it from the genomic study. So genomics is divided into two parts. One is structural genomics and the second one is functional genomics. The structural genomics includes whole genome mapping, which has genetic mapping using some genetic approach and physical mapping using some physical mapping approach. Mapping says it is the same as a map, like globe is the map. In globe, you have the information about India is situated at what place, which country is present in the north of India, south of India, and then east and west of India, where the England is situated, where the Hind Mahasagar and Arabian Sea is situated. These are map which you shows the different the position of different countries, rivers, mountains onto the globe or onto the map. Simultaneously, we talk about the mapping in the molecular biology term. It will show you what is the position, what is the size of the chromosome, and at what position your particular gene or marker or centromere or telomere is present. So that is mapping we have to do either through the genetic mapping or physical mapping. Then once the mapping is done, we can go for whole genome sequencing. Once the whole genome sequence means what? Adenine, guanine, cytosine, thymine. Only ATGC, ATGC, ATGC will be present. Now the question is arising that if you go for the human genome sequencing, you will get ATGC, ATGC, ATGC. Rice genome sequence also ATGC. Microbial genome sequence also ATGC, ATGC. So what is the function of this ATGC, ATGC? From that ATGC sequence, the random sequence of ATGC, which is present on different chromosomes, we have to find out what is the function. So there are some bioinformatics approach which is very quick and of course very much in use. Based upon that, you can minimize your effort. You can go by bioinformatics approach. You can get some information, and those information you can collect, confirm from transcriptomics analysis, proteomics analysis, and metabolomics analysis. Whatever approaches are available in the laboratory, based upon that, we can confirm the total function of the particular identified gene. So for that, we have the different genomics tool, that is genome annotation and bioinformatics. What is genome annotation? That is obtaining biological information from unprocessed sequence data. So see here, they are the ATGC sequence. From this ATGC sequence, whatever information, molecular information, physiological information, genetic information, cellular information, whatever information we can gather, we can collect from ATGC, that is called genome annotation. And how we can collect those information? Based upon gene homology. We know that I will start from very simple example, that glycolysis cycle, where the glucose is converted into glucose 1,6 biphosphate, right? Glucose is converted into glucose 1,6 hexokinase enzyme is involved. Now, if this particular hexokinase enzyme must be having some protein sequence, and it must have derived from some gene sequence. Suppose you have done the sequencing of a microorganism, or you have done the sequencing of a plant or animal, and some of your sequence, 2 kb of your sequence is 95% matching with hexokinase gene. We can predict and we can say that this particular portion belongs to hexokinase gene, and then you can reconfirm through the different molecular biology approach, right? So based upon sequence based feature, what are the sequence based feature, we will study in the next slide. Gene prediction using FGenish and other genes software. There are several other software which is available for the gene prediction analysis, right? Again, this is not improving. I don't know why it is a problem. It is not moving. Slides are not moving. Yes, FGenish is the perhaps I should spend little less time. FGenish is AB initio gene prediction. Now you can say that how the software can say that this is a gene. We all know that what are the gene specific features. We all know that what are the gene specific signature. कैसे हम कहते हैं कि ये एक जीन है? इसलिए कि gene is starting from start codon, gene has a stop codon, gene has a splice site, gene has polyadenylation site, gene has cap site, data box, stop codon, promoter site, etc., etc. So this software is prepared in such a way that wherever all these informations are available in a particular sequence, the software will say that it is a gene. But what is the function of the gene? There are several other software which is available online. These are repeat masker, blast and gene finder, blast X, half-wise tRNA, repeat promoter, tRNA, pseudogenes, prosite, pfam, blast, pasta, etc., etc. There are so many software available. I'm giving just a few examples that once the gene is identified, once the software will say that okay, this particular portion of the genome is having a start codon, a stop codon, polyadenylation site, cap site, etc., etc. This is having gene. Now you take that gene sequence and find out what is the function of that gene, that particular gene, using the other software available online. So once you identify the other software with the help of other software, then you can go for the gene function through some experimental analysis. You see here, this is the International Rice Genome Sequencing Project. These are the physical map for IRGSP. We have taken the all clone one by one. We went for genome sequencing. After that, we did for gene prediction. And after prediction of the gene, that is called genome annotation, subcellular location of the protein, domain analysis, GC content, physical mapping, etc., etc. That has been done after the genome sequencing is over, right? So this is using the rice genome sequencing genome information. We have identified that there are several 195 genes present on chromosome 11. We were interested in chromosome 11, 195 disease-resistant genes we have found, and we also found that there are several locations where the genes are present in clusters. So here you see the 16 resistant gene analog cluster, 10 cluster, 21 cluster, 30 clusters. So these clusters shows that the single gene is not working alone. They are influenced by the other genes, which is activating or deactivating the genes present, the genes in question. The classification of disease resistant genes, there are 5 classified basic classification of disease resistant gene, that is detoxifying enzyme kinases, NBS-LRR, that is nucleotide binding site and leucine rich repeats, and extracellular receptor and receptor kinases. These are the examples. It is identified detoxify enzyme identified in maize HM1 gene, tomato PTO gene, Arabidopsis RPS2 gene, tomato CF gene, and rice XA21 gene. Right? These are identified from using the different pathogen, that is maize HM1 gene using the pathogen Copiolobus carbonum. So these are the name of some pathogen, against that it is identified, and these are the different function of that protein, HC toxin, serine, threonine, protein kinase, etc., etc. I will not spend much time on this slide. I'll go ahead. Once you take the genome sequence and once you go for gene prediction, this is the dialog box of F Genish gene prediction site. You have F gene. Once you open the F Genish gene prediction site, this is the dialog box. You insert your sequence, the genomic sequence, and then after that, here is some option that whether this gene belong to plant or animal or microbe. You have to do that, and then you click on the search button. This software will tell you that how many number of genes are present in the given sequence. So this is my result. It will show you the coding sequences. It will show you the where is the polyadenylation site, how many genes are there. So based upon my analysis, we found that there are out of 195, there are NBS-LRR gene 80 number, leucine rich repeat protein kinase, NBRC, serine protein kinase, etc., etc., serine serine protein kinase. So these are the different signature of disease resistance gene. So I was interested in disease resistant gene. You may be interested in glycolysis gene, or somebody else may be interested in stress or drought tolerance gene, or zinc and iron transporter gene, etc., etc. So based upon that, you can give the option and you can select that what are the genes available on that particular the genome sequence which you have provided. These are the different the disease resistant genes which is identified on chromosome 11, XA21, XA4 these are the positions of chromosome 11 where this gene sequence is present on the chromosome 11. PI18, PI7, PI44, these are the blast resistant gene, and these are the bacterial blight resistant gene. So my lab is basically working on bacterial blight resistant genes. As we have seen in the previous slide, that there are a huge number of clusters of genes are present on chromosome 11, and same situation is there for the other chromosomes also. This shows that a number of additional genes are involved in R gene, that is resistant gene mediated defense activation. This suggests that existence of multiple resistance signaling pathway. Since the one gene is not completely independent, they are dependent upon the other genes present in the vicinity, whether it is reported or not reported. If we silence the other gene, the main gene function will be affected. So this shows that one gene is involved in multiple pathways. So the rice genome information that 37,500 genes are there, to identify the function, to confirm the function it may take 1 decade, it may take 1 century, right? So because huge number of molecular biology experiments are required to confirm the predicted information. I have taken that 195 chromosome 11 disease resistant gene, and we have also done the comparative analysis of chromosome 11 disease resistant gene with other chromosomes of rice, chromosome 1, 2, 3, 4, 5, 6, 11, 12. So we found that maximum approximately 50% of the chromosome 11 identified disease resistant genes are matching with the chromosome 12. That chromosome 11, 12, it is reported that they are duplicating chromosome with each other, right? And then of course I have checked the genome sequence, the gene sequence of 195 with rice unigene database and with rice est database. More than 80 and 90% homology I found, right? And then I did the comparative analysis of Nippon variety in sequence, that is Japonica variety with Indica variety. We found that more than 90% the disease resistant genes present on Japonica variety of chromosome 11 of rice is matching with Indica chromosome 11 of rice. Then we have done the comparative analysis with the other cereals like wheat, maize, barley, and found that it is matching with wheat. Approximately 100 genes are out of 195, 100 genes are matching with wheat. And we also did the Arabidopsis and tomato, etc., etc., brassica tomato. We found that with Arabidopsis it is matching little significantly, but not with the other dicots. right? So there are different diseases. I have less time. I'll try to finish within 10 minutes. Is it okay? 10 minutes, Dr. Abdul Halim Saab?
Sure, sir. You can, sir.
Okay, okay, thank you. So there are different diseases caused in the rice, caused by bacteria or nematodes or virus or parasites and fungus also, etc., etc. So these are the some common disease in rice: bacterial leaf blight, blast disease, sheath blight, narrow brown spot, sheath rot, false smut, brown spot, Tangro viral disease, bacterial leaf streak disease. Students, you must have learned about some of these diseases in your graduation, in your intermediate also, and you will be learning more in your masters and in your PhD program about the disease, about the progression of disease, the mechanism, and the disease resistant genes identified against all these diseases. Right. So these diseases, several groups are working on this disease throughout the world to increase the rice production. My lab is basically working on bacterial leaf blight. So bacterial leaf blight is caused by bacteria called Xanthomonas oryza. It causes a reduction in photosynthetic area and 1,000 grain weight. That empty grains are there. Up to 20 to 50% yield loss is there. But once this disease occurs, such type of pandemic like corona pandemic is going on, if such pandemic is there in plant, then up to 80% the grain production is there. You see here why we are concentrating upon rice, because rice is the staple food for more than half of the world population. And as we know that the world population is increasing, this red color is the world population. So world population is increasing day by day, but in that context, rice production is not increasing in that context, right? So it is our responsibility to find out what are the reason that rice production is not increasing, and we have to increase the rice production. By the year 2035 we need an additional 116 million tons of rice, according to United States Department of Agriculture report, right? And Indian subculture, there are huge number of people and children die because of hunger. So you see here that rice is less, roti बहुत कम है, लेकिन रोटी मांगने वाले और रोटी की जरूरत महसूस करने वाले बहुत ज्यादा लोग हैं. So it is our responsibility to increase the yield production. If we control the bacterial blight disease, then automatically to some extent we can increase the yield also. And several other groups are working on development of yield. We will see how these things are working. So the coming to the functional genomics approach, these are exploitation of available genetic resources. There are some gene available in the gene bank, that is in the different species of Oryza, which is responsible for disease resistance gene. Presently, there are 23 species of Oryza identified, and this genetic resources is the hub for valuable gene crops. There are several wild variety, right? There are several wild variety available in the world, and these wild variety has a lot of genes, but these genes are not identified so far. If they are attacked with bacteria, their disease symptoms are not occurring. That means they must be having some gene which is controlling their disease symptoms, but grain is not there. So we can identify those genes from the wild variety. These variety, even they are infected, they don't show the infection symptoms. So if we identify that why they don't show infection symptoms, so what are the genes responsible for that? If we isolate these genes and transfer these genes, either transgenic approach or plant breeding approach, to the popular rice variety, then the chances of the rice production will increase. Till date, there are 40 resistant genes known as XA genes, that is Xanthomonas oryza genes, and some defense-related genes are identified in for bacterial blight. Reports of breakdown of the disease-resistant genes, for an example, XA4, XA1, XA21, etc., etc. So my lab is basically working on allele mining. What is allele mining? Allele mining is the identification of some of the gene present in the wild variety. So as I told you, there are several wild variety. Grass, जो जंगली राइस है, उसमें डिजीज सिम्टम्स नहीं आते, डिजीज उसमें डेवलप नहीं होते, राइट? दैट मीन्स कि they are having the huge number of genes. We have to identify that huge number of genes, and because they belong to grass family, it will be easy for us to transfer those genes from wild rice to the popular rice eating rice variety. If we are successful, then there is a possibility that we will be decreasing the disease symptoms, we will be decreasing the infection symptoms, and we will be increasing the gain also. So identification and isolation of novel and favorable alleles of agronomically suitable genes from the crop gene pools to suitably deploy for the development of improved cultivar. So this is our aim. And I'll just show you the example. Suppose in one variety, one XA gene is identified. This is this red color I am showing here. That this gene is identified. This gene is known for disease resistant gene. Now, my lab is basically looking for whether the same gene is available in any other wild variety or not. If it is available, what are the similarities and differences among those genes, and what will be the final effect of those genes under for the disease progression also, and for the yield progress also. So suppose the yield is here for this actual gene. We identified allele 1, allele 2, 3, 4, 5, 6, 7, 8, and we found that the second allele 1, the production is little more than what is actual gene is identified. The allele 2, the production is little less. Allele 3, the production is almost double. Allele 6, production is very less. Allele 7, again, little improvement. So among all these gene pool, one variety we have identified, a wild variety, in which the disease progression is less and yield production is double than the actual gene. So we can identify that why the production is doubled. What are the similarities and differences between this gene and this gene? At what sequence level there are differences? We can say that this particular gene is superior to this gene. So based upon this superiority, we can isolate this gene, identify the function, and use this gene for the plant breeding and transgenic approaches. So this bacterial blight, I'll not go into the detail. It is caused by bacterial blight disease caused by Xanthomonas oryza. It infects the xylem of rice plant, causes lesion of leaf, and eventually plant death takes place. Once the pandemic occurs, up to 80% yield loss is there, right? So that is zoo family, that is Xanthomonas oryza, secretes several factors which causes infection as well as trigger host resistance. Once they release some of the secretion factor, then the XA gene family is activated. And once the XA gene family is activated, if the plant is having disease resistant gene, they will combine with these factors and they will not allow the bacteria to grow inside the plant, and they will be resistant, and there will be no effect on productivity. Whereas the plant which does not contain disease resistant gene, it will unable to fight with the secretion factor of zoo, and then the plant will be susceptible. There are some conserved domain, that is leucine rich repeats are available. XA21 is one of the gene which contains which is GC rich region, 233 base pair region, which is common to all XA families. XA3, XA27, these are the examples. Both are single gene. XA13 is a recessive gene, and these genes can be used for the for making for breeding or transgenic approach for making the plant disease resistance using the allele mining approach. Allele mining is one of the method as I described in my previous slide. These are the different steps which we are using in the laboratory. We are collecting the germ plasm from throughout the global level. Of course, I am not collecting, but I have collaboration with the Directorate of Rice Research. They are collecting these germ plasm. These germ plasma are being grown, and in the either in the field or in the greenhouse. After growth of particular growth, we go for the infection analysis. After infection, we see that out of suppose 100 plants we have infected, we try to see that how many plants shows the disease resistance and how many plants shows the infection symptoms. Those plant which does not show infection system, we isolate the DNA from both the plant, and then we do the gene based primer. We do the PCR analysis. After that, we take the PCR product, go for PCR product sequencing, and then go for sequencing analysis and expression studies to find out how this particular gene is different from the previously identified gene. And based upon our analysis, we go for selection of novel alleles and we identify. In short, the allele mining approach belongs to the steps are selection of target trait, then identification of accession with desired phenotypic trait, that is the trait which does not show disease symptoms. We target the gene, we go for the PCR amplification, then sequencing the finding of, then go for sequencing and then find the variation. So there are two basic approaches for allele mining. One is sequenced based allele mining, and second one is tilling based allele mining. We have discussed about sequenced based allele mining. The tilling based allele mining, I don't want to go into the detail. But there are some procedure after that. Once the sequence variation is there, then we go for the comparison of the sequence data with phenotypic data and identification of superior allele. Once we identify superior allele, we can say that this allele is better than the previously identified allele. Based upon that, we have taken some of the variety Oryza nivera, and these are the different accessions of Oryza nivera. These are the different Xanthomonas isolates which we have collected from different parts of the India, and then we go for the PCR analysis. These are the PCR product. If we get the PCR product different from the control one, then we can go for sequencing, and then we go for sequence analysis, and then we find the transition and transversion after then single nucleotide polymorphism. After that, we see that what is the difference between the original DNA sequence and the newly identified DNA sequence. You see that this is the resistant variety. The first one is a resistant variety, and this particular DNA sequence is not available in the other sequences, other variety. So that means we can say that because of this particular region, because of this addition of 3 nucleotide, this particular variety is very resistant, and other variety is very susceptible, right? So simultaneously, we have found several other differences of XA5 gene, XA21 gene in different parts. Then we have also done the expression analysis, and with before infection we have isolated RNA. After infection we have isolated RNA, and we found that there are some differences of expression false before infection and after infection. That means some genes are activated, and this is showing because of those. You see here in this variety, the disease symptoms is starting from top till here. Here also at least 2 or 3 centimeters are there. But in this variety, it is very less. In this variety, the symptom is very less. In this variety, the symptom is very less, right? So this is the overall allele mining procedure. Allele mining, the application of allele mining also deals with the allele identification, allelic variation characterization, allelic synteny, similarity analysis, evolutionary relationship analysis, promoter mining, etc., etc. So this whole functional genomics approach includes genomics, proteomics, and transcriptomics. So you have the in metabolomics also is coming into the picture. GCMS, LTM, NCMS, FTIR, and several metabolomics approaches are there. Based upon those approaches and metabolomics, we identify some molecules, and we can say that these molecules may be responsible for some genes, may be responsible for disease resistance, some protein may be responsible for disease resistance, and some compound may be responsible for disease resistance. Now it is our responsibility to correlate these compounds with genes and proteins and find out whether there is some correlation or not. The finally, coming to the summary, that we have identified total 195 disease resistant gene on chromosome 11. Out of that, 140 were present on the long arm. So gene identification, clustering, and annotation provided a wealth of information for disease resistant gene. We have identified I succession conferring high disease resistance gene. Two resistant variety from XA5 alleles were identified in Oryza sativa cultivars, and presence of single dominant XA21, XA27 alleles among the studied accession illustrate the selective maintenance of the gene. So that's all my presentation. This I was part of the rice genome where Dr. N.K. Singh, Dr. Mahapatra, Dr. Tia Sharma, Kishore Gaikwad, and all the rice genome sequencing group people were there. He is Dr. Professor P.S. Srivastava was my PhD guide at Jamia Hamdard. And here some of the data I have provided you from Anirudh and Vimalata was my PhD student. I am having collaboration with Dr. R.M. Sundaram and D.S. Laha and Dr. Girish at DRR. And of course, the funding is available in my lab from DBT, DST, and UGC. So that's all my presentation. Thank you very much for your kind attention.
Thank you very much, sir, for your nice presentation. We had in our curriculum about disease-resistant genes on not specifically on rice, but we have a bit in one of the paper what we are teaching for MSCs, where we have got a lot of information which is you are working live working. You are working on the same concept, so it is very excellent work, and it is the work which has opened our eyes, and we feel that our scholars who are attending this conference will have an have a glance, and they will learn so many things from you. Very kind
Sure,
of
sure. I
very
will
kind of
be
you,
very
and
happy.
Department of Botany and entire faculty, and the cast and crew of our department is expressing heartfelt thanks for your early morning presentation. on rice. Rice, though it is a staple food of, as you told, more than half part of the world, especially Asian subcontinent, is more dependent on rice for the staple food. Uh, Thank you very much once again for your nice presentation, sir.
Thank
If
you.
queries come, we'll be sending you to the mail what you have given to us. Now I request our next speaker. As next speaker from Mumbai University is having technical difficulty in joining, can I request Professor Altaf Ahmed sir to start his presentation? Altaf sir,
Okay, okay.
sir, you can start your
Yeah,
presentation.
yeah, I will start. Just,
You start sharing your screen, sir. Shortly,
yeah,
I'll be giving your
please
introduction.
wait for 1 minute.
Yes, sir. Sir, your screen is visible. I'll start your introduction, sir. Short while I'll be finishing the introduction. Wait, sir. Wait a
Okay.
minute. Professor Altaf Ahmed, sir, is professor in plant biotechnology at Department of Botany, Aligarh Muslim University, Aligarh. He earned his PhD in botany from Jamia Hamdard University in 1988. Subsequently, he worked at Indian Agriculture Research Institute as research associate. He then joined Jamia Hamdard as assistant professor in botany. At Jamia Hamdard, he established a state-of-the-art molecular biology laboratory in the Department of Botany and worked on the identification of candidate genes for low nitrogen tolerance in wheat and rice through analysis of proteome profile, and expressed and differentially expressed miRNAs. In 2008, he was a visiting scientist at Virginia Polytechnic and State University, USA, on biotechnology overseas associateship of the DBT, Ministry of Science and Technology, Government of India. He got advanced training on the development of FRET-based genetically encoded nanosensors for real-time monitoring of amino acids. He also visited in University of Freiburg, Germany on INSA DGF fellowship in 2013 to study differential expression of root-specific 9-sulfate transporters genes of popular by real-time PCR. Dr. Ahmed monitoring of various metabolites in living cells. He's also studying the proteome profiling of nutrient efficient and nutrient inefficient crop plants. He published more than 125 research papers and edited 8 books. Dr. Ahmed has presented his work at several national international conferences, including Third International Crop Science Congress 2000 Germany and 14th International Plant Nutrition Colloquium 2001 Germany, 10th Congress of European Society of Photobiology 2003 Austria, International Conference on Reduced Nitrogen in Ecology and Environment 2006 Austria, 4th International Nitrogen Conference 2007 Brazil, and International Botanical Congresses 2005 and 11 at Austria and Australia. A regular viewer and on the panel of editorial boards of national and international journals, Sir is also a member of important national and international scientific societies. He guided thesis research of 30 PhD students, 12 as a supervisor and 18 as a co-supervisor, and over 30 MSc students. He has organized several national and international seminars and symposia. Sir has handled several research projects funded by various agencies like DST, CSIR, UGC, PCRUM, ICAR, INSA, BBSRC UK, and UKRI GCRF UK. He also worked as deputy coordinator of UGC SAP program
Speaker.
project of the Department of Botany, Jamia Hamdard. Based on his research contribution, he was honored with INSA Medal of Young Scientist 2003 and Professor LSS Kumar Memorial Award 2003 of Indian National Science Academy. Young Scientist Award 2005 of CSIR, Council of Science and Technology, Government of Uttar Pradesh, and elected as Associate of National Academy of Agricultural Sciences. He also received Young Scientist Award of Academy for the Advancement of Agricultural Sciences, New Delhi, conferred by then Honorable President of India, APJ Abdul Kalam. With this introductory part, I welcome Sir to give his presentation. Good morning, sir. You can start your presentation, sir.
Thank you very much, Dr. Haleem. Very good morning to all of you. I'm Altaf Ahmed from Aligarh Muslim University. So I would like to present before you some of my work on nanosensors. So we have developed some nanosensors that are genetically encoded, and they are really required for the measurement of metabolites in living cells. We know that a large number of plants and animals genome have been sequenced so far, microRNA have been sequenced so far. So the primary goal of in the post-genomic era, before the scientists, is to assign the functions of each of gene encoded by a given genome and their integration in the metabolic and regulatory network. For the assignment of the function of the gene in the metabolic network, various omics are being used like transcriptomics, proteomics, and metabolomics. Like we know that all the genes are present in the coded form in the genome, and they are transcribed in the form of mRNA by transcription process, and they are then translated in the form of proteins, and then the proteins, which are mostly enzymes, catalyze the function of catalyze various biosynthetic pathway to make various metabolites, and they make the metabolic network. One new approach has been now developed, that is fluxomics, that is really required for the regulation of the gene in the metabolic network. Because so far we know various genes are present in the metabolic network, but how they are regulated, at what step they are regulated, are not very well known. And this fluxomics approach is being used to understand the regulation of the flow of regulation of the gene in the metabolic network. And it is really a flow of metabolites through a metabolic network in living cells. If you see the transcriptomics, it is an approach in which the expression of a gene at the transcript level has been studied. And it is a simple approach. The tissue is taken out, total RNA has been isolated, and out of the total RNA, mRNA is purified. This mRNA is now reverse transcribed into cDNA. Then after adapter ligation, cloning amplification, the sequencing is done, and raw data is obtained through the bioinformatics. We can identify a large number of genes which are expressed at a given time at a given site. And various tools are available for this transcriptomics approach, like Northern Blotting earlier tool, quantitative real-time PCR, microarray. These are high throughput tool. RNA-seq is again a high throughput tool. Proteomics approach, in the proteomics approach, what we do, the protein is isolated from the tissue, and it is separated by using IPG strip on the base isolated focusing point. Then they are run in the two-dimensionally by SDSPs, and we found a differential expression of the protein when we compare the control one. Like some protein are overexpressed, some protein are induced, some proteins are absent, some protein are underexpressed. So four kind of condition we obtained after doing the proteomics analysis in the gel. When these differentially expressed protein are identified, they are excised from the gel, and they are sequenced through the MALDI-TOF or LCMS, and the data is obtained. Data is now used to find out the gene which are really coding that kind of protein by Mascot software or Swissprot. Various tools are present for proteomics, like flow cytometry, Cytov, 2D page, very well known, protein macro and MS. In the metabolics approach, what is done, again we have to take the tissue, and the samples is now grinded or is processed. After that, the metabolites are separated by various chromatography approaches like GCMS, LCMS, and then by mass spectroscopy, the metabolites are identified and data is analyzed by various softwares. The tools are available for metabolomics study or HPLC, GC, GCMS, LCMS, and NMR. So in all the three approach, common thing is that we have to take the tissue. Tissue is now processed to isolate the mRNA or protein or metabolite. So we have to process the tissue. We have to damage the tissue. After that, fractionation is done and analysis is carried out. But in case of fluxomics, the approach or the study of the metabolite has to be done in the intact living tissue, and that is a tough task. If you want to, even from the beginning, take, if the nutrient is uptake by the plant from the root, that is to be done by intact plant. If the nutrients or metabolites have to be translocated from one place to another place, that has to be done in the living plants. And even in the metabolic network, in the cell or in the suborganelles, the metabolites move from one step to another step. That is to be done in the living cell and intact cell. So for that, the key problems in the young field of fluxomics are concentration of a particular molecular species in a physiological environment cannot be determined. It is not possible to visualize how the concentration of metabolite varies across an organ, tissue, and cell. And there is no way to detect how metabolite level changes in response to the environmental stimuli. And the change in the metabolite level cannot be monitored in real time in living cells. So these are the key problems in the fluxomics. These are not the problem in the transcriptomics, proteomics, and metabolomics. But the need of the day is that we have to find out the gene which are regulatory in the metabolic pathway. And for that, fluxomics approach is being used and is developed. So for doing these achievement or to finding to address these questions, we need to have a tool that can determine the concentration of particular molecular species in a physiological environment. We need to have a tool that can visualize how the concentration of that particular molecular species varies across an organ, tissue, or cell. We need to have a tool that can detect how metabolite level changes in response to the environmental stimuli. And we need to have a tool that can monitor these changes on the metabolite level in the real time. So earlier approaches have been used, like a German group developed non-aqueous fractionation technique, and in that, but that was invasive, in which we don't need to crush the tissue, but it has very little cellular resolution, and it is very sensitive to artifact. Later on, spectroscopic method like NMR imaging and positron emission tomography were developed that can provide dynamic data, but again, the resolution is very poor. The most advanced technique is the development of single cell shape sampling method, in which a microsyringe is developed, and this microsyringe take the sap from the cell in the living cell, and you can now take the metabolite and identify the metabolite by different methods of a living cell. But it can only give you a snapshot of a metabolite level at a given time in the given tissue. You cannot do the time-lapse experiment. So none of these methods enable real-time measurement of the metabolite in living cells. Thread-based nanosensors are the ray of hope. These are the devices which are non-invasive and enable real-time measurement of flux of metabolite in cellular and subcellular compartment. How they are developed? Very simple way. In very simple way, they can be developed. Like we have to take the protein, fluorescence protein. We have to use the Fred phenomena. Then we have to take some protein which are especially peripheral binding proteins. So fluorescence protein were identified, characterized, and by these three scientists, Shimomura, Shafi, and Shine. They got Nobel Prize by the discovery, identification, characterization, and first analysis of the green fluorescence protein from the Pacific jellyfish. Later on, some proteins were identified from other organisms like sea anemone. And these proteins are now successfully used for the sublocalization of the various organelles, various parts, like BFP is being used for mitochondria, mCellulin is used for actin, tubulin is used, AGFP used for tubulin, YFP is used for Golgi, penicillin is used for MKO, M3 is used for histone H2B. So EGFP, green fluorescence protein, also has some variant for some variant like BFP, blue fluorescence protein, is developed by replacing the tyrosine at 66 position by histidine. Uh, Theorin at 65 position was developed by replacing this with the serine. ECFP was developed by replacing tyrosine or GFP at 66 position by tryptophan. YFP was developed by replacing the threonine 65 or GFP by glycine 65. So these are different variants which are available nowadays in the market for different purposes, for localization, for visualization of the metabolites or protein-protein interactions. Second phenomena is the Fred phenomena. It is a process in which a donor fluorophore in excited state non-radiatively transfer energy to an acceptor molecule. So how it is done? Combination of two fluorescence protein shows the Fred. It is well known now. Like CFP, cyan fluorescence protein is a donor fluorophore. It has excitation at 430 nanometer and it has emission intensity 520 nanometer. Yellow fluorescence protein is acceptor fluorophore. It has excitation at 512 nanometer and emission at 535 nanometer. So when we put these proteins at very near, the distance is less than 10 nanometer, then the emission intensity of the CFP excite the YP. So now you can see by exciting the CFP, you can get the emission from CFP as well as YP, and this is the Fred. So the emission of the CFP when the two fluorescence protein are very near, the distance is less than 10 nanometer, then the Fred phenomena is shown, Fred process is taking place. So energy transfer non-radiatively from one fluorescence protein to another fluorescence protein. But there are three conditions, at least three conditions are there for Fred. Number one, the emission intensity of the donor should overlap excitation intensity of the acceptor. So there is no Fred if there is no overlap between the excited emission and excitation intensity of the donor acceptor fluorophore. Second one is the distance between the two fluorophores should be less than 10 nanometer. And third one is there should be correct orientation, that is they should be at the parallel orientation. Third component is periplasmic binding protein. There are a large number of such proteins are available in the gram-positive bacteria, and they are used to take up the nutrient different metabolites from outside environment. And these proteins work like a Venus flytrap. Like in the Venus flytrap, there is a hinge. The two lobes are joined together with by using a hinge, and when the insect binds sit on the any of the lobe, it closes. So these periplasmic binding proteins work like this. This is periplasmic binding protein that I shown schematically. When the metabolite is bind, it shows its conformation like this. So our approach is that we have to search a suitable periplasmic binding protein for a particular metabolite. For that, we have to make the nanosensor. What we had CFP at one end of the periplasmic binding protein. We had YFP at another end of the periplasmic binding protein. When we excite the CFP and there is no metabolite, then you will not see the fluorescence from YFP. The emission only CFP is taking place because the distance between the two are more than 10 nanometer. So there is no Fred. But in the presence of the metabolite, there is change in the conformation of the periplasmic binding protein. And at this time, when we excite the CFP, we will get the emission from CFP and YFP both. So Fred occurs. So it clearly shows that presence of the metabolite, and we can quantify the metabolite by doing the titration. You can see the clear shift in the emission intensity to both CFP and YFP. So here is without addition of the metabolite. This is CFP peak. This one is the YFP peak. And after addition of the metabolite, the CFP peak is lower down and YFP peak is increased. So the emission intensity of the CFP is lower down. That gives the energy to the YFP. And this is the Fred. The ratio of the YFP and CFP is calculated, and we can quantify.
Sir, your slides are not moving.
It's moving now, but in my one it is moving. Your screen is paused. So I am now resume share.
Yeah, now it is moving, sir.
It's moving. Okay, okay. Somebody has started sharing that one, I think. Uh, so, have you seen this one, this approach in the slide moving?
Yes, yes. Pink color,
Okay.
periplasmic binding protein.
Okay. So this is a binding protein. We add the metabolite there, then the conformation is changed. So the approach is that we add CFP at one of the one end of the periplasmic binding protein, and we add YFP at another end of the periplasmic binding protein. And at this time, when the CFP is excited, there is emission from CFP only. There is no emission for YFP because the distance between the two fluorescence protein are more than 10 nanometer. But when we add metabolite or the metabolite is available in the cell, so the conformation of the periplasmic binding protein is changed and the two fluorescence proteins come together. And when we excite at this time CFP, we can get the emission from CFP and YFP both flat occurs. So in this way, we can identify the metabolite, we can identify the concentration of metabolite by knowing the ratio of the CFP and YFP. So here is the spectrum of the CFP and YFP emission intensities. You can see without the metabolite addition, there is a peak of CFP here, which is larger than the YFP peak. This is the YFP peak. But after addition of the metabolite, the peak is changed. This is the CFP peak, which is lower down than the earlier one, and this is the YFP peak. So ratio is calculated. If we see the ratio earlier it was 0.8, now it is 2. And we can quantify the metabolite after doing the titration. This is the titration curve of the YP and CFP ratio after addition of different concentration of metabolite. So we can get the KD affinity of the metabolite of the sensors by measuring the metabolite. So how it is done? So when we see the change in the CFP and YP ratio, the ratio is calculated. So here is the picture. We perform the experiment in the cell, and after excitation of the CFP, we can get the Fret ratio. This Fret suppose is 2.3. Then this is deduced corresponding ligand concentration from the in vitro calibration curve that we make earlier already made, and this is now we can calculate the concentration of metabolite in the cell. Suppose it is 0.2 millimolar. So on the basis of the already made calibration curve, we can quantify the level of the metabolite in the cell and the change in the metabolite level during the different time intervals we can calculate because it is in the living cells. So how it is changing, at what time, how it is changing with the treatments, we can identify the level of metabolite without damaging the cell, without this is purely non-invasive. So first sensor was developed by Rogerson, that is chameleon sensor. It is very widely used. They use calmodulin, and they added CFP and YFP fret pair, and concentration of the metabolite calcium is measured in the in vivo cells by using this sensor. Later on, a Fromer group in the Stanford University developed 3 more sensors: maltose sensor, Philip mol, glucose sensor, Philip glucose, ribose sensor, Philip ribose, and they are very successfully used in the identification of the different maltose, glucose, and ribose in the living cells. My lab had developed several sensors like glutamine sensor, GLINH, leucine sensor, live-K, methionine sensor, methane, zinc sensor, glycine betaine sensor, sensor for lysine, and sensor for vitamin B12. So we developed leucine sensor using the live-K protein for E. coli, and we added the CFP and YP in both the end, and this is the designing of that sensor. We developed methionine sensor taking the methane protein from E. coli again, E. coli K12, and we added CFP and YP both the end, and this is the methane protein. We develop lysine sensor by taking LOA BP protein from E. coli again, and we added CFP on both the ends. Vitamin B12 nanosensor was again developed by taking the B2F, BTUF protein. Glutathione sensor we developed, it is recently published in scientific report, YEEB protein from the Salmonella typhi. Recently, we developed ROS sensor by taking the oxygen transcription factor, and this sensor is also we developed recently. So how we develop the nanosensor? I want to give you an example by taking the nanosensor for lysines. What we do first, we identify the suitable protein. We go to the protein database and search for suitable prepared binding protein. Like we have identified LAO, lysine binding protein from Salmonella enterica typhimurium, and we found this is the pocket. This one is the pocket for the binding of the lysine, and here you can see clearly after binding of the lysine there is a change in the conformation of the this lysine binding protein LAO. This is the open form, this is the closed form, so clear conformation change can be seen. So amino acid sequence are taken out from the NCBIs, and nucleotide sequence are taken out from the NCBI, and we design the sensor on the basis of the sequence of the CFP and LAO BP sequence and YP sequence, and these are the restriction sites that we use to ligate different genes. So genomic DNA was isolated from Salmonella, that is taken from IVRI Jatnagar Bareilly, and we design in-house nano primers by and I did the Hindi third side and SP1 side in the forward reverse primer, and we got a fragment after amplification 741 base pair. After that, we also amplify the CFP and YP by suitable restriction site like BAM-H1 is added in the forward primer, and Hindi third was added with the reverse primer, and the size of this CFP is 700 base pair, and we got exact 700 base pair fragment when we run it in the agarose gel. YFP was also amplified, and we added the SP1 site and SEC1 site on the forward primer and reverse primer. And the size of the YP is also 700 base pair. And after amplification, we run the amplified product, and we got a fragment of 700 base pair. These three amplified product of gene of interest, CFP, YP, were cloned into PGMT EG vector by TA cloning. These are the PGMT CFP, PGMT YP, and PGMT LAO. They are separately were cloned into PGMT. After that, they were validated after extendation. LAO was validated, and we cut it by Hindi third, and double decimal also by Hindi third, and we got clear validation after extendation. CFP again was validated by restriction by taking the BAM-H1 and BAM-H1 and Hindi third pair, and we got the expected fragment that we have expected after before digestion. We also validated the YFP cloning by using the extendation process, and in this way we have done single digestion by SEC1, and we did the double digestion by SEC1 SP1. So after validation of all these three amplified product and their successful cloning, we have ligated all the three. So LAO was sandwiched between the CFP gene and YP gene, and again cloned into PGMTG vector. Validation was again done by resting digestion, and this time we have did single digestion by BAM-H1 and double digestion by BAM-H1 SEC1, and clear fragment, expected fragment was obtained that shows that the cloning and ligation of these CFP, LAO, and YP successfully ligation in PGMT vector. After successfully validation of these chimeric genes, CFP, LAO, and YP, we have settled it into the PRZ vector, which is a expression vector. And beauty of this vector is that it has his tag. It has his tag here. This his tag is used to purify the protein. So the whole construct CFP, LAO, and YP was settled to PRZ vector, because we have to express the protein, we have to characterize the protein for that. This was again confirmed by restriction digestion, And this time we did the single diacetyl by Sec1 and double diacetyl by BAM-H1 and Sec1. And we got the expected fragment that shows clearly that the construct has successfully transferred into PRZ-B vector. After that, we whole construct was sequenced through outsourcing, and we got 99.9% similarity with the designed construct. So after these two level of validation, we move further, and the protein was expressed into E. coli BL21 because it is in the now PRZ-B vector. This is the whole protein PRZ-B, CFP, LAO, YFP, and the cell were grown and the induction was given. After that, the cell were harvested by ultrasonication, and the sensor protein was purified by using the nickel anti-histag affinity chromatography, and we got the purified protein. And this purified protein was kept overnight at 4 degrees Celsius so that the protein, the sensor protein get its native conformation. So this histag, I showed you that PRSET has the capability to add histag with the uh, express protein. The protein, the purified protein was again checked by running through running it at SDS page, and we got a single large expected band of the protein, that is a sensor protein here. After isolating the protein of the purification of the sensor protein, in vitro characterization of the nanosensor was carried out. First, we conducted the spectral analysis, emission spectral analysis of the sensor, and we see here that it is done from 430 nanometer to 440 nanometer to 580 nanometer, and this one is the peak of without the addition of the lysine. You can see here the YP, CFP peak. This one is the YP peak. But by the addition of the lysine, there is shift in the CFP and YP peak. CFP peak is lower down and YP peak are increased. So it clearly shows that fret occur. This sensor is showing fret. So it means there is a clear conformation of the protein, the clear transfer of the energy from donor to acceptor fluorophore. We also now check pH stability of the sensor, because in the cell there different cell have different pH. So our sensor should be stable for different pH. So we conducted pH analysis. We conducted the fret analysis at different pH, and we found that the sensor is stable 5.5 to 8.5 pH, very much stable. There is no change in the fret. We again check the specificity of the sensor, because in the cell different kind of similar amino acids or similar metabolites are present. So we took arginine, glutamine, cysteine, lysine, and leucine to check the specificity of the sensor, and we found that the sensor very specifically monitor the lysine, and there is no change by the addition of arginine, glutamate, cysteine, and leucine. So it shows that our sensor is very specific for a particular amino acid, that is lysine. After that, we have calculated and where we have conducted the experiment to calculate the affinity of the sensor. So for that, we did the titration of the sensor protein at different level of the lysine from nanomolar to molar concentration, and we found that the sensor can measure up to 97 micromolar of the lysine very easily, the KD 97, and this sensor is named as Philip K. Because the level of the lysine can increase or decrease beyond this 97 micromolar. So it will not work at higher millimolar concentration. It will not work at nanomolar concentration. So to solve this problem, what we did, we have made affinity mutant of lysine nanosensor by through the docking study. We identified that there are several amino acids that can be replaced or substituted by different other amino acids to make the more affinity mutant of the sensors. Like we found that tyrosine at 40% can be replaced by alanine, arginine at 77% can be replaced by leucine, phenylalanine at 52% can be replaced by alanine, asparagine at 161% can be replaced by alanine, isoleucine, serine at 72% can be replaced by alanine. So by we have designed the primers to do these experiment, and by site-directed mutagenesis kit, we develop different mutants by replacing these amino acids by other amino acids. And we successfully found that there is a clear change in the affinity of the nanosensor. Like this nanosensor is Philip K 142 micromolar. This sensor is 150 30 micromolar. Wild type is the black one, and this pink one is the can measure the lysine in millimolar concentration. So here is the table that can be seen very easily that wild type nanosensor can measure between 14 to 980 micromolar lysine. Philip K30 mu can measure the lysine from 4 to 208 micromolar concentration. Philip K142 mu can measure the lysine between 20 to 1000 micromolar. Philip K306 mu can measure the lysine between 65 to 1500 micromolar, and Philip K14Q can measure the lysine between 2 to 150. So these are different affinity mutant for different for measurement of lysine at different levels. After that, we conducted the in vivo experiment to know whether this nanosensor can perform the monitoring of the lysine in the living cells. So first we conducted the experiment in the bacterial cell, and after transfer of the sensor in the bacterial cell, we added the lysine, and this one is the control one in which there is no lysine was added, but this fret is showing the lysine is already present in the bacterial cell. So you can clearly see that addition of the lysine, the fat ratio is start to change after 5 minutes, and it get the saturation at 30 minutes. So sensor is clearly monitoring the level, increased level of the lysine in the living bacterial cell. So here you can see that without damaging the cell, without disrupting the cell, we can monitor the lysine level in the living bacterial cell. We also conducting again specificity analysis test. So this is the PBS buffer. There is no fret. This one is the lysine clear monitoring the lysine in the living bacterial cell is done by the fret by the sensor. We added the arginine. There is no change in the fret. The only thing is added. There is no change in the fret. The additional histidine. There is no change in the fret. So again, it is showing specificity in the living system. This one is the confocal imaging, CFP, YFP, and this is a MERS one. So sensor is clearly expressed in the bacterial cell through confocal imaging. It can be seen. So after transformation of this, after studying the monitoring of the lysine by using nanosensor, we move to the eukaryotic system, that is yeast. So we settle the construct into the PNT vector. So in this case, we use the gateway technology. So first we have transfer the construct CFP, LO, IP into the PNT vector, and after the PNT vector, we conducted the gateway cloning. We use PYS dash vector, that is a yeast expression vector, and we settle this whole construct in the PS dash vector. After that, this whole construct, this vector that is containing the sensor was transferred into the yeast, and you can see here that yeast the sensor is very well expressed in the yeast cell also. This one is the image of CFP, this one YP, and this was merged cells. And then we also we have seen that the sensor is expressed in the cytosol of the yeast. There is no sensor expressed in the vacuole. This is CFP, YP, and merged cells. And then we monitor the level of the lysine after addition of lysine. What is the change in the emission intensity of CFP and YP? You can see here that this is the emission intensity of CFP. And the lysine is added at 60 seconds, and you can see that the emission intensity of the CFP is decreasing continuously. And after addition of the lysine, the emission intensity of YP is increasing continuously up to 325 seconds. Fret ratio was monitored, and fret ratio between by using CFP and YP ratio, CFP and YP emission intensity. So after addition of the CFP at 45 seconds, the increase in the fret ratio until 255 seconds. So this also clearly shows that sensor works well into the eukaryote system also. And in this case, uh, 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 there is no damage of the cell, there is no processing of the yeast cell in the living intact cell. You can monitor the level of the lysine by using this nanosensor. After that, we transfer this nanosensor into the plant, Adosis theliana, and the sensor was first shuttled to the PLD gate 100 vector, which is a plant expression vector. And here the sensor is also expressed. This is CFP, YFP, and merge cells. So this is the system through which we monitor the level of the metabolite by taking the confocal microscopy and perfusion system. In this syringe system, different concentrations of the lysine were or metabolite were added, and here is the controller system that is controlled the by different units. And here the solution moves one by one, and here is the exit of the solution, and here we put the camera through the microscope and monitor the level of the metabolite through Fred ratio. So this is the whole system. uh, Confocal microscopy, rotative microscopy,
Sir, your voice is stuck up, sir. Hello, sir.
range. Resolution is subcellular again, cellular resolution again, minimally invasive. It can do the monitoring the living cells. It can do the real-time monitoring. MRI, the sensitivity is mid micromolar to millimolar. No subcellular resolution. Yes, cellular resolution is there. Minimally invasive. Living cell in living cell can be done. In real time, it can be done. PET, the sensitivity is 1 to 40 becquerel millimolar square. No subcellular resolution, no cellular resolution. It is not minimally invasive. It can do the actually it can do the monitoring in the living cell. It can do the real-time monitoring also. X-ray synchrotron, the level is 1 milligram per kg. No subcellular resolution, no cellular resolution. It is not a minimally invasive. It cannot do the monitoring living cell. It is not real time. So these are the most advanced techniques, and the nanosensor, which is very, very low cost, can do the subcellular resolution monitoring, minimally invasive living cell, and real time monitoring. Advantages are that it is non-invasive. It can be targeted to subcellular organelles like in Golgi body, mitochondria, chloroplast, in any subcellular organelles you can target, and it can allow real-time imaging. So the problem that we have seen in the fluxomics in the initial slides. So this sensor can solve these problems. Like it can determine the concentration of a metabolite in physiological environment. It can visualize how concentration of that particular molecular species varies across an organ, tissue, So these sensors are a good candidate for fluxomic studies, and that study that help in the studying the regulation of gene in the metabolism. Thank you very much for your kind attention.
Sir, very kind of you for such a detailing on very latest technology of FRET. So I'm very kind, and on behalf of the Department of Botany, Telangana University, I express my deep sense of gratitude towards accepting our invitation at short notice and giving such a beautiful and wonderful presentation. Being a plant biotechnologist, you have gone the heights. What we can't we can't even assume. You have reached such a modern science. Classical botany has become such a wonderful subject by you, sir. You have turned mm, milestones of our classical science. So with these few words of your kindness, I would like to request the next speaker, Dr. Ahmed Ali, sir, to start sharing his screen. Dr. Ahmed Ali from Mumbai University. Dr. Ahmed Ali, Please start sharing your screen. Or to Aruna ma'am. Aruna ma'am, please introduce speaker. Aruna ma'am, over to you. Ma'am, unmute yourself. Ma'am, unmute yourself, ma'am.
Good morning. Good morning. Sir, am I audible, sir? Am I audible, sir?
You're audible, ma'am.
I deem it as a privilege to introduce today's speaker, Dr. Ahmed Ali, who is currently working in the Department of Life Sciences, University of Mumbai, as assistant professor. Earlier, he worked in the National Institute of Pharmaceutical Education and Research, that's NIPER, and he studied at Jamia Hamdard and University of Mumbai, obtaining his MSc and PhD degree in biochemistry. He has over 13 years of teaching and research experience, and presently is heading the molecular biochemistry laboratory in the department. His areas of research are protein and DNA biochemistry, with special contributions on glycation of biomolecules, DNA damage, and anti-glycating properties of natural products. He also made significant contributions in the area of artificial sweeteners and their role in the process of glycation. Cyanobacterial systems are another thrust area in his lab, where researchers are exploring various applications of these organisms in the field of cosmetics and health benefits. He has received several extramural grants from government and private funding agencies, and he has collaborators from national, international laboratories. He's serving as reviewer and member of editorial board of various international journals like Chemosphere, Frontiers in Plant Sciences and Microbiology, International Journal of Biological Macromolecules, Annals of Phytomedicine, and other journals of Elsevier and Springer. He has contributed more than 25 research articles in peer-reviewed international journals. He is also author of one book and several book chapters from Springer and Elsevier publishing houses. He has presented his work in many international and national conferences. And with this brief introduction, I now welcome and invite Dr. Ahmed Ali sir to present his deliberation on the topic nutritional and health benefits of spirulina. Thank you, and over to you, sir. Thank you.
Sir, check your audio. Ahmed Ali sir, please check your audio. Your voice is not reachable. Sir, no, your audio is not clear, sir. No, sir, your sound is not there. No, you're not audible, sir. Sir, your audio is not clear, sir. You're not audible.
Yes, sir. Can you hear me now?
Yeah, अब आ गया आवाज आपका जी.
Okay. Thank you, sir. Thank you thank you, Dr. Abdul Haleem sir, for giving me this opportunity and the department to invite me to deliver a lecture. And we just heard one of the pioneers in plant biotechnology, Dr. Altaf Ahmed. He was my senior in Hamdard, and then I'm in regular touch with him. And I admire him for his research work. So with this, I'll go and is my screen visible now, sir?
Yes, your screen is visible.
Yes. So as discussed with you earlier, I'll be delivering a lecture on this nutritional and health benefits of spirulina, which I have worked during my PhD also. I'll be sharing most of the relevant informations available on this organism. So it's a photosynthetic multicellular cyanobacteria, which also looks blue-green in color because of the pigments like chlorophyll and phycocyanin present in this organism. It is one of the very wonderful organisms as it can be grown in fresh water as well as seawater. There are more than 20 different species. Out of that, two are very prominent, S. platensis and S. maxima. A lot of work has been done on these two organisms. This has been grown naturally and native in Africa, Asia, and South Africa. Now it is being grown all over the world. It has been also used as a model organism to study the mechanism of nitrogen metabolism because of its very high protein content, because it also shares the nitrate assimilation pathway like higher plants. It has been also exploited for its bioremediation potential to remove the heavy metals and prevent the accumulation of nitrate and phosphorus in water bodies. And one of the very important feature of this organism is that it has been rated as GRAS, generally recognized as safe by the US FDA, because the genes which are responsible for biosynthesis of toxins are absent in this organism. So if you see under the microscope under 400x you can see this organism looks like a helical structure. And for a botanist, this classification is very important. It belongs to phylum Cyanobacteria and order Oscillatories, and the genus is Spirulina. And in literature, we also find people write it as Arthrospira. So some of you might have seen this bottles in the market, which are commercial application or commercial products of this organism where they sell spirulina because of high protein content and other features which I'm going to discuss in this lecture. The spirulina flakes and powders which are available and which are sold commercially are also available in the market. So what some organizations and institutions say about this organism, it's world's most powerful food. It is also the most ideal food for mankind, as recommended by United Nations. USFDA said that spirulina it's food for the future, and in 1974 it was declared by the UN World Food Conference that it's best food for the future. So there are some advantages that it can be grown in the freshwater, as I mentioned in the previous slide. Women have taken this as a entrepreneurship in Chennai or that area. So 1 to 3 grams of spirulina per day for 6 to are enough to rehabilitate a malnourished child. A lot of work has been done to use this as organism for food and feed. It requires very less space related to amount of protein produced, 15 times less than sugar cane, 20 times less than soybean, and 25 to 50 times less than rice. It requires very little water as compared to other plants and organisms. The high yield is also very high. So these are some of the literature which we have published in our lab. This is a book chapter. This is a review article where we have talked about the potential of this organism in nitrogen metabolism as well as bioremediation, and some of the works which correlated this organism as for its high protein content. So we found that the activities of nitrate assimilation enzymes are very high as compared to rice, which is a known low protein content organism. So this is the summary of what the nutritional or biochemical analysis of this organism has been reported. It's one of the most high protein content organism, almost reaching to 70% by dry weight. There are many minerals present in this. We also find most of the vitamins in this organism. It is also rich in carotenoids, pigment, C chlorophyll, as well as phycocyanin. The one thing that it's a very, it has low amount of sodium, which is required for by many people. So if we see the numbers, it appears to be promising as nutritional supplement, but high amount of this may prove to be toxic, as the reports have suggested. So the WHO report suggests that the protein content of this was 57% carbohydrate or this, and what makes it a very interesting organism that it contains almost all the essential amino acids except histidine, and we can see that some amino acids are in very high content like glutamic acid, aspartic acid, and so if you see the FAO standard protein recommendation, we can see that this organism contains almost all those amino acids which are required for in our dietary supplement. So it can fulfill the daily requirement of protein content. Another very interesting feature of this organism that it has a very high content of gamma linolenic acid, which is the precursor of many of those prostaglandins and leukotrienes. So it is also a very good source of many of these fatty acids or essential fatty acids. You can see alpha-linolenic acid, and omega-3 fatty acids. So the biochemical analysis of this organism also show that it is rich in many of these vitamins, so it can be used for the food as well as feed. Why I'm using that, it has been being used for feed for animals as well as in the fishes. So if you can see that it's also rich in most of the minerals, which are sodium is quite low, So the report says that okay, it is low in some species and high in some. Another very important aspect of this organism that it does not contain phosphatic polymers and phytic acids, which limit the bioavailability of iron. So it can be used for eradicating the iron deficiency and contain most of the essential minerals in an amount as required for daily intake. So if we see the literature, we find that many of these bioactive compounds, other than what we have seen, vitamins and other things, these uh, bioactive compounds are there, which can be sterols like antimicrobial agents, gamma-linolenic acid can be used for the synthesis of prostaglandins, arachidonic acid is there, sulfolipids are there, which can be used for the, prevention of HIV or retroviral disease, vitamin A for source of vitamin A, vitamin phycocyanin as a source of pigment as well as antioxidant activity. So these are there. So we can see the summary of this that it has been used for as the source to prevent many of diseases. So if we summarize that, what are the benefits of taking 5 to 10 grams of spirulina every day? But I recommend here that please refer the established research reports instead of reading through the many websites which give some kind of wrong information also. So it increases the CD4 count, which helps in the prevention of HIV. It also it's also rich in beta carotene, contains vegetable protein which can be easily digested, and high concentration of B vitamins. It's also an excellent anti-inflammatory source for arthritic patients, and contains anti-aging properties, improves digestive health, and because of the absence of those phosphatic acid and other things, iron can be easily absorbed. So some of the health benefits which are reported in the literature on the basis of clinical and pre-clinical studies. These are anemia, antibiotic effect, cardiovascular disease, cardiac toxicity, neurogenic disorder. Uh, Spirulina has also found as a tool for genetic studies as animal feed, and people are also exploring this organism for biofuel production in the pharmaceuticals production of and pigment for production from spirulina for textile and other industries there. So what we have done in the lab, I'll just briefly mention here that we are working on one aspect of diabetes, that is glycation, which is basically an interaction between the sugar and free amino groups. So the amount of glycation products which are harmful increases during the hyperglycemic condition. So the two stages are early glycation and late stages of the formation of glycation products. And this late stage or the combination of these compounds which are harmful are known as advanced glycation end products, which have been implicated in many pathophysiological conditions like diabetes, cataract, retinopathy, and neuropathy. And so basically, this ages they inhibit or alter structures of molecular proteins or DNA-induced DNA damage or cause oxidative stress. So this scheme shows how this sugar protein interact. So this is a non-enzymatic interaction which leads to formation of cis base, then amadori product like fructosamine, and then advanced glycation product. Some of these structures are shown here, and they cause So some of the parameters which we have measured in the lab include the spectroscopic studies and some protein structural alteration. Some of the studies I'm sure showing here that it increases the amount of browning. Browning is is a basic parameter which tells us about the amount of advanced glycation and products generated during this interaction of glycation between the sugar and protein. And we also see this browning in our day-to-day food cooking, where the food turns brown because of this heating of the food, where carbohydrates and proteins interact. And this is also a concern for the food industry. So we can, I have used the model as the BSA as the protein, and glucose here, G is the glucose here. And we can see that the browning has been reduced in the presence of spirulina extract. So similarly, the early glycation products were also reduced in the presence of this extract. Then we measured the advanced glycation product, that is protein carbonyl content. As the amount of advanced glycation products increase, we see an increase in the protein oxidation. You can see here the carbonyl content was reduced by more than 55%. Another feature, another parameter which is measured in the lab using spectrophlorimetry is the measurement of total advanced glycation products. So this is done at 330 and 410 nanometer of excitation and emission respectively. So we can show here that the amount of advanced glycation products or total agents of spirulina extract. Then we analyze the DNA damage which is induced due to the protein, due to the glycation, or in turn generation of free radicals. So we can show here that in lane 2, the DNA, the plasmid is completely damaged in the presence of glycation system. So on the other hand, if we add the spirulina extract and other cyanobacterial, we can see that there is no damage to the DNA, or the damage has been prevented or reversed. We also did a brief study on the preparation of skin whitening effect of this organism. So we prepared the skin cream using some of these available chemicals as well as cyanobacterial, and we checked the properties, physical properties of this. It was adhering to the standard guidelines. Then we measure the antioxidant activity of this. We can show that extract had this and cream had this kind of activity. So we did a FRAP assay also. One of the ways by which creams are measured for their biochemical feature is that whether they prevent the tyrosinase enzyme, because tyrosinase enzyme is involved in the formation of the pigment. So we can show that kojic acid is a standard for this, and we also check the orange peel extract which has been used traditionally in our household. So we could show that cream which we produced in our lab was quite efficient in preventing the activity of tyrosinase. So that means it can prevent the formation of melanin pigments, which will help in the whitening of the skin. We also did the anti-inflammatory activity. We measured the protein inhibition assay. We used the diclofenac, which is the standard for this inflammatory assay, and we could show that cream was more efficient in preventing the proteinase activity. So with all these studies, I mean, we are still we have to do the animal studies and other parameters to establish the efficacy of the skin and its safety. So with this, I can conclude that it's a very good source of nutrient, has amino acids, minerals, vitamins, antioxidants. Uh, it has antibiotic resistance to as well as prevention of cardiovascular disease. So it has been shown to be quite useful or significant in the prevention or treatment. And what we have done is that we have checked the anti-glycation property of this extract, which we could see or report because of the ability to prevent glycation at early and late stages and prevention of glycation-induced strand mix and or break in the DNA. And we also show that the extract had cosmetic properties and cream was prepared, which we could show that it has skin whitening as well as anti-inflammatory, antioxidant, and anti-aging properties. So this some of my students have worked on this project and the fundings which I have received from various sources. So thank you for your patience and
Thank you so much, sir, for your presentation. As we are running short of time, I express deep sense of gratitude towards your active participation in our international conference and making our event successful. So,
thank you, sir.
a big
Thank you,
thank
sir.
you, sir, for you. Now, I request Dr. Jalandhar to introduce our next speaker, Dr. A. Sabita Rani, Madam, from Department of Botany, Usman University. Over to you, Dr. Jalandhar.
Okay, sir. Thank you. Very good morning to all of you. It's my immense pleasure to introduce Dr. A. Sabita Rani.
Wait, Jalandhar, wait.
Madam.
Madam is found. She was made co-host and now she is not appearing.
Dr. A. Sabita Rani is presently working in Department of Botany.
Wait, she is not there in the meeting.
Okay, sir.
Actually, she was there in the morning, but now she is not found. Dr. Swati. Oh, Savita. Dr. Jalandhar, you can start because madam is joining.
Okay, sir. Okay. Dr. Savita Rani is presently working in Department of Botany, Usmania University, Hyderabad. She obtained her PhD in genetics from Center for Plant Molecular Biology, OU, and MSc in Botany from Kakatiya University, Warangal. Postdoctoral fellow at Indian Institute of Chemical Technology, that is IICT, Hyderabad, under DST Young Scientist and DST Women Scientist programs 2001 to 2007. She has published 47 research papers in national and international journals, contributed 8 book chapters, and presented 49 papers in national conferences in India and international conferences in USA, China, Bangladesh, Sri Lanka, Egypt, Kuwait, etc. And she has organized 4 seminars and 2 conferences, 2 workshops. And she has completed 4 major research projects funded by DST and UGC, and 1 minor research project. And she has guiding many doctoral students, and 4 students they awarded their PhD under the guidance of madam. She has worked as a member Animal Ethical Committee for Gandhi and Usmania Medical College, Hyderabad. She is actively involved in science communication programs, resource person for many training programs, jury member for DST Inspire programs. She science lecture for Vignan Prasar, teleconferences and video lesson in biology. She was elected as a fellow of Telangana Academy of Sciences in 2017. Distinguished Scientist Award here in 2018 by Genesis Urban and Rural Development Society, Hyderabad. Gurnani Award for Best Oral Presentation by Indian Women Scientist Association in 2019. Her research interests are, she's interested areas are plant biotechnology, bioevaluation, plant secondary metabolite production through tissue culture. Thank you very much, sir, for giving me this opportunity. And now I welcome A. Savita Rani, madam. Over to you, madam.
Ma'am, you start. Ma'am, you start sharing the screen, ma'am.
Yeah. Hello, good morning. Is it audible, sir?
Yeah, you are audible.
Okay. I'll just, so, yeah. Just 1 second. Is the screen is visible, sir?
No, not yet screen is seen.
Now?
Now it is coming. Yeah, it has come.
Just thank you very much for the nice introduction. First of all, congratulations for the organizing committee and Department of Botany, Telangana University for conducting this wonderful international seminar, and also giving me the opportunity to share some of the information which I'm interested. Okay. So the topic of today's presentation is biotechnological approaches for in vitro production of secondary metabolites for sustainable utilization of the medicinal plants. So we know very much like medicinal plants are the important ingredients of the majority of the traditional as well as the modern system of medicine. And the India is the rich biodiversity country. We know that it is one of the 17th World Megadiversity Center with the various 15 agroclimatic zones, and it is a rich source of the medicinal plants, around 17,000 to 18,000. So out of this, already there are more than 1,000, approximately 960 species are used in the trade as well as the industry. So there is a lot of rich biodiversity in the India. So whatever the latest advancements in the medicine, still 45% of the world's population depends on the traditional medicinal and herbal preparations. So out of this, 10% of the prescribed drugs and 25% of the common medicines which comes from the plants. So there is a lot of demand, the market potential for the medicinal plants around 2 lakhs crores. That is the market potential of the medicinal plants, and in which the India contributes around 2,000 crores of rupees of the medicinal plants. Though we are rich in the biodiversity, still our market potential is very less. Probably the reason is majority of the medicinal plants what we are using is collected still from the wild, and not many medicinal plants are cultivated worldwide. So that could be the reason of the not fully exploiting the medicinal plant trade or market. Okay, so if you're coming to the medicinal plants, the part which you are using may be a different, maybe it is a root, maybe it is the stem, or if you, for example, if you are looking into that, this is the diagram, if you can look into that, you can see most of the percentage of the
Ma'am, your sound is not audible. Ma'am. Savita ma'am. Savita ma'am. Vidya ma'am, what to do as madam is not available? Shall I ask next speaker to start? Okay, Dr. Srinivas. Can you introduce Dr. Swati? Dr. Swati, are you ready?
Yes, ready, sir.
Swati ma'am.
Yes, sir. Yes, sir.
Yes, you share your screen, ma'am.
Okay.
A great good morning to one and all. Myself, Dr. Devara Srinivas, Department of Botany.
Swati ma'am.
Yes, sir.
Screen share change, ma'am.
Okay, sir.
Department of Botany, Telangana University. I would like to introduce our guest speaker of this session, Dr. G. Swati Madam, GDC Putturu. Dr. Swati Madam is an assistant professor in geology. She had good research bent of mind with it is a multidisciplinary approaches. Her research work mainly focus on neurological disorders, Parkinson's disease, epilepsy, Alzheimer's, with identifying potential herbal plants, a protective agents. She had also studied hepatoprotective activity with combination of herbal plants and also identifying potential active agents. She published 6 books and 32 articles in journals of national and international repute and presented papers in many seminars. She had been invited to write editorial Hidden Tale of Pollution, Need of Solutions in Current Trends in Biotechnology and Microbiology. She is advisory board members in World Journal of Pharmaceutical Research and Pharmacology in Cambridge Scholars Publishing Limited. Editorial board member in Journal of Natural Products Research Updates and associate editorial board member in Current Trends in Biotechnology and Microbiology, CTBM. Thank you for giving me this opportunity to introduce the speaker, Dr. G. Swati Madam. Thank you, one and all. Over to speaker, Dr. G. Swati Madam.
Thank you, sir. Thank you for your introduction, sir. Thank you, sir. Am I audible?
Yes, madam.
Okay, sir. Thank you. This topic is very interesting from the age-old when the human civilization has started, the new dimensions in understanding aging. I'm Dr. G. Swati, Assistant Professor, Zoology Department, Government Degree College, Puttur. Now we are going to see how the human civilization has struggled to defeat death and not go to the process of aging. You can see in almost all mythologies, like in Norse mythology or in Chinese mythology, we had a fountain of youth. And if you go to the Hindu mythology, Shiva trilogy or like that, there will be so many ideas like immortal life. So even Jesus has said, "Who have eaten my flesh and drink my blood has eternal life." This is also shows that we are trying to seek the immortality. In ancient Greek myths, ambrosia, the food or drink of the Greek gods, is often depicted as conferring longevity or immortality upon whoever consumed it. It was brought to the gods by the Olympus by dove. So in all the mythologies, every mythology, and even in Mayan mythology, Yucatecan mythology, every mythology has how to achieve immortality, how to defeat death, and how are we going to not go to the process of aging, which is inevitable. So in this long run, as the human civilization has started about 2 lakh years ago, now we are going to achieve some small new inventions through the scientific methods, which we can defeat this aging process, which we can slow down the aging process. So can we rewind the clock, or can we reverse the aging process? So this is our interesting topic from the age old. Now we are going to see how we can do all these things. So are there any longest living organisms till now? I mean in the earth. So we can see, if you see the sea urchin, this has the longest average lifespan about 200 years. It is a small echinodermata which can live up to 200 years. And even the koi fish is living up to average life of 226 years. And long life in eel and macaw, African elephant. Actually, these African elephants usually live up to 80 years, 60 to 80 years, but this African elephant has an average lifespan of 152 years. And we know about the tails of Galapagos giant tortoises, which has a heavy average lifespan. Even this bowhead whale, which is a mammal in the ocean, has an average lifespan of 200 years. Even Greenland shark is having the age of 400 years. Ocean quackhead, and also we have some of the immortal living organisms. So can we achieve this immortality? I mean, not immortality. Can we defeat the death? This is a very interesting topic, and we can see this rotiferan deloids has a lifespan about 100 million years. It's nearly an immortal, and even the lungfish Neoceratodus lives up to 100 plus years. A glass sponge is living about 15,000 years above plus. So there are so many are living organisms which are really becoming the immortal and are also slowing down their age and are defeating the death. So these planaria larvae, rough-eye rockfish, sea anemone, zombie bugs. There are so many organisms which are living very long. They are like, if you see any organism, the age is related to the size or something like that. There are so many big animals also which are living, some more bowhead whale or green land shark like that. Okay. So now this is a very interesting animal, naked mole rat. This is living up to 35 years plus. Actually, the rodents of this family are living only for 4 to 6 years, but we will expect about some 6 years or 7 years. But in one of the labs of Buffington's lab, it is now still living 35 years plus. And you know, the thing is, it lives in subterranean grounds, and it is when the infection comes or when any predator comes, it will die. But it's normally, it doesn't die like other rodents of this family. And also you can see the naked mole rat has a queen which reproduces, which is fertile till its end. Actually, this immortality or aging process in 4.5 million years ago, when the earth has started, and 3.7 million years ago, when living organisms has coming rise on this earth, these organisms were first of all like living immortals only, as they will be cloning only. So they had given a prize for evolution, that is mortality. So to become immortal, they had to evolve. They become immortal. So they had given this prize. So now again we are trying how to get to the immortality. I mean, without defeating the death. Yeah. This is a water bear, first animal to survive in space. Its lifespan is about century. This is also a water bear. So what is aging? So when we observe a person who is in front of us, we expect that who is aging. We as gray hair, memory loss, wrinkles, and brittle bones. These are some of the things which we observe when we talk about the age. So irrespective of this, we had so many neurological disorders also. As we age, as we run through the age, we are accumulating all the diseases and also all the disorders also, because if we go on, if a mission or anything is running out, we can know that it will be having wear and tear off. Like that only, these neurological disorders are the most significant things. When the aging starts, they will be accumulating, like Alzheimer's. Alzheimer's is the major disease, and dementia, epilepsy, stroke, and Parkinson's disease. Parkinson's disease is where the dopamine neurotransmitter will be decreasing in the substantia nigra. So beta amyloid plaques will be accumulated in Alzheimer's disease. Like epilepsy, acetylcholinesterase will be increased. It's like that stroke is the major thing where the age related problems will be coming. Here we can see the aging is related not only to neurological physiological things, but it is related to neurological diseases also, like hypothalamus mediated aging progression. The underlying cellular mechanisms for hypothalamus mediated aging progression comprises dysregulation of nutrient sensing. So everything will be altered, altered intercellular communication, stem cell exhaustion. Even in some experiments, they had proved, some of the scientists have proved that by injecting the stem cells into the hypothalamus, again the older mice have returned to the, again returned to like young mice only. Epigenetic alterations are so many things which are related to the aging process. Yeah, this is the everyone knows that the ends of our chromosome is protected by telomeres. You can see here the ends of the chromosome. Now the thing is, if we go on use, if you go, there is a half-life limit for every cell. That is, after 60 times the cell ceases to divide. Okay, then actually these telomeres will be going. The ends of the telomere caps will be shortening. Like if you see, if you use a candle, if the candle goes on burning, what happens at the last? It will be ended. So these telomeres are also one of the factors in aging. So if you can see here, the length of the telomere when it started, when the cell started proliferation, it will be long. And when when it goes on divide, the length of the telomeres will be shortened. So if you see here, eukaryotes and prokaryotes, what is the difference between the telomeres? If the telomeres is that the thing that if the telomeres are long, the aging process can be stopped. We are thinking, but in aging and cancer, limited lifespan, because due to the presence of telomeres, sexual reproduction complex bodies are also because of this telomeres. Absence of telomeres, only the clonal growth will be there. So if you take our lobster, green spotted lobster, this is one of the immortal organism. I mean, it is nearly an immortal organism where the telomerase will be adding on its telomere ends. So we can see only it will die because the shell of it will be overgrown and it will compresses the visceral organs. So the visceral organs will be compressed and it will die. So in our in new studies also, our human race has came to many solutions. Like in Tel Aviv University of Israel has recently published HBOT chambers where hyperbaric oxygen treatments can reverse the process. In this also they had identified that the telomeres length will be as much increased in this process, like 38% will be the telomere process will be increased. And they had seen in 35 adult healthy adults were 64 above the 64 years old, and in those tests they had those research they had significantly discovered that HPOT therapy, I mean like hyperbaric oxygen treatment or in hyperbaric chambers, the telomere shortage can be increased, and also amount of senescent cells. Senescent cells will be as the cell goes on aging, some cells won't die, as we know that apoptosis is there, a programmed cell death. After some limit, the cell will die, but some cells will continue to live again like that only. So while they are living, they are called senescent cells or zombie cells. While they are living, they will be also, I mean, nearby cells will be also damaged. Nearby living cells are also will be damaging. So these senescent cells can be removed. If the senescent cells are removed from the area, maybe the cells, that place will be taken by the new cells, which can divide again with an apoptosis program. So, but this is not a holy grail. But we are thinking that telomeres shortening is considered to be the holy grail in biology of aging. But in a Canada research area, telomeres is said that over the past year they dispelled the theory of longer telomeres equal to longer life. But up to now, we are thinking that we came to a conclusion, but again we are going to the starting point only, because the longer telomeres won't give to longer life. For example, jellyfish. Jellyfish doesn't have a longer telomeres, so but it is living. It is an immortal organism. So now we are also having we are going to another solutions like clotho-protein. Clotho, a natural human protein, has a great potential to delay the diseases of aging. So Aging process means accumulation of diseases. Accumulation of all the diseases is known as nothing but aging. So by delaying this aging process, we can defeat even the death also. So this protein is also had came to now existence, like loss of cognition, kidney failure, diabetes, cancer, all the things that are believed to be associated with body's production of this protein. So if you see in this research of Kuro et al., which was published in Nature 1997 and Curso et al. In Science 2005, they had shown that Clotho knockout gene 80% lifespan, shorter lifespan than a normal mice. Even in normal mice, I mean, when the Clotho protein is normally produced, it was like here. And when the Clotho protein was overexpressed, again the lifespan has increased about 30%. 30% of the lifespan has been increased. So we also have some drugs like Richard Miller has found 4 drugs like rapamycin, acrobose, 17-year alpha estradiol metformin. So what about this rapamycin? A drug which block the certain kinds of cancer. It has increased 20% of lifespan in both male and female mice. These are experiments. These are all going on experiments in mice. Somewhere even in dogs also they had worked out, and not even the human trials had started. But this rapamycin is given as a drug for a cancer patients, some different kinds of cancer patients. Now thought to be increase the lifespan in mice, like both male and female mice. To the normal patients, it's unknown. I mean, normal healthy persons, it's unknown. This can keep the age-related diseases at bay and extend the span of life. And next one is acrobose. Acrobose, this controls diabetes. This controls diabetes, promises anti-aging effects. This is the one which promises the anti-aging effects, and it works only in male mice. Male mice, it increases 22% of the lifespan, and in females it increases only about 5 to 10% lifespan in mice. And now we can see this 17 alpha estradiol. It is also one of the promising drugs which lengthen the lifespan only in male mice about 15 to 18%. There is no effect in female mice. In metformin, this is also used as a treatment for diabetes. Now it can show the anti-aging effects. So another candidate has also came into this defeating the death or slowing down the age, that is NAD+. Now the research is also going on NAD+, nicotinamide, you also know coenzyme. So as we age, the NAD+ levels will be decreased. If you can see here, as we age, it's gradually decreasing. The NAD+ levels are going to decrease. So are there any sources? So why NAD+? Why NAD+ levels? What is the use of NAD+? So it is contributed to DNA damage. So it can, it won't do DNA damage to telomere attrition. So everything we can see here, it is also related to NAD+. So this is also going to a good candidate. So how can we get NAD+? We can get by 2 forms, NAD+ pure form or NMN pure form, which can be converted again into when we take NMN tablets, it will go and convert it into NAD+. This is nicotinamide mononucleotide. And also we can get good levels in naturally available food. Older mice fed with NMN, they had the same levels of NAD+ levels of younger mice. So this is a very nice candidate. So now Yamanaka factors. Yamanaka factors, the Nobel Prize in Physiology or Medicine was awarded jointly for Sir John B. Gordon and Shania Yamanaka for the discovery of this Yamanaka factors. These Yamanaka factors will make a mature cell again into a stem cells. So stem cell, when injecting before we were talking like when we inject a stem cells again into some researchers have found that when injected into a old mice, the stem cells into the hypothalamus, it was gaining again like the new younger mice. So now these Yamanaka factors were related to mature cells turning over into the stem cells. So this is one of the results of Yamanaka factors. David Sinclair, now who was the famous scientist in gerontology, I mean, in this aging research, Harvard Laboratory has published that it was 3 out of 4 Yamanaka factors will again set back the methylation clock, Howard methylation clock, turning functioning of tissues back into the stem cells. The same study offers evidences to support the hypothesis that the epigenetic clock is a lethal driver of aging rather than an adaptive response to the damage. You can see these OCT4, SOX2 and KL4 genes of these 3 factors were used as a model tissue in eye. They can in a glaucoma old mice, and the retina and axon has been regrowth. So not only in AWS, we had seen 2 candidates. Now third one is regular fasting. So regular fasting is also could lead to the longer and healthier life. In a 2000 study, if we talk like this, we need a scientific proof. So many scientists had proved that in 2017, a study showed alternate day fasting was an effective daily calorie restriction for losing weight and keeping in pounds off. This was done by Trepansky, Jeff et al. 2017. He said that losing weight or fasting alternate days may lead to the longer and healthier life. In also in some studies also in 389 4.4 years followed for in 4.4 years 389 years routine fasters with their intermittent fasting, they had survived without some diseases like cardiac arrest like that. Whether new intermittent fasting weight loss regimes led to the longevity requires further study by Benjamin Horner in 2019. And also calorie restriction, that this also gaining to improve our longevity of life. In the study, Blemonte and his colleagues compared that who ate 30% few calories with rats on a normal diet. The animals' diet were controlled for 18 months to 27 months. In humans, this would be roughly equal to 50 through 70 years. So the rats who had ate 30% fewer calories than the rats who ate normal calories, they had lived more than them. Like 18 months to 27 months, they had been increased. In a study of scientists from US and China had provided that most details report to the date of cellular effect of a calorie restricted diet in rats, while the benefits of calorie restriction have long been known. The new results how this restriction can protect against aging in cellular pathways was published on Cell on February 7th. So there are not only the factors like aging, biological, physical, social, psychological, social, environmental, and economic determinants were also included in this a process of aging. So if you think that psychological, if you see the Parkinson disease, those who aren't laughing, who are not laughing well, they will be easily getting this Parkinson disease, because at the onset of 40 years or 50 years, gradually the level of dopamine will be decreased. If you laugh well, the dopamine levels will be stabilized. So this is one of the main culprit for getting Parkinson disease. There are many culprits like genetic, and also the use of, when we are using all out left, there will be a rotinone and everything, which will induce the neurological diseases also. We are continuously accumulating those things, and eventually we are going into the neurological diseases. In social also, actually the aging candidates about 50 years or something, they will be socially eliminated. 60 years above, they will be retired from the works and they will be eliminated. So when the brain activity and the brain has been decreased, all the cells will be decreased. So these things are very mainly including. There is no one thing which gradually decreases, slows the age. We can see the people who are working very hard and who are with the socially
Excuse me, Dr. Swati,
comfortable
your
zone.
screen is not moving.
Sir?
Your screen is not moving. All things need, that one is coming. All things need to consider, that is coming. You are
Okay, sir. Before that slide, it's not coming, sir.
gallery restricted. Okay, thank you has come.
Okay.
Okay, this last slide. Okay.
Yes. Okay. Not only the social things, but also environmental things like pollution. Pollution triggers the ROS mechanism in human beings, like reactive oxygen species. So when the reactive oxygen species is, they are more accumulated in the human body, intent they will damage all the tissues which are nearby. So if the damage is going to be done more and more when we are exposed to pollutants in the roads and everything, ultimately the age will be increased, because we need to take more and more antioxidants to scavenge them. But that is not possible, because in this busy world who are running hard, they can't take. So the aging process is not going to slow down for them. And also we can see actually the human civilization has achieved this aging goal also. Now the average age has been raised. In US it was 80, and I think in India it also now it is going to rise about 65 years plus. In India it is like, in some societies it is above 65, in some societies it's 60, like that. I mean, according to the economic determines that they were there. And also in the new studies, like when we are talking about the food healthy diet or NMN or B3 diet, and also like that, when we are consuming more by probiotics and everything, they are also going to help us to stimulate whatever the ROS things are accumulated in the body. They will be scavenging out, and also all the nutrients which we eat will be easily available to us. And also we can see economic determinant is a main theme which is determining everything, psychological, physical or psychological or social or environment. Everything is goes on to this economic determinants, because anything we are dependent is on this only. If we had a good wealth, then we will be okay, then we are not worried. If we had low wealth, we'll be like worried. If we are going on worried, neurological disorders will be crept onto you, and the aging process will go on. Like if the stress is going on, again, if the stress will be acting on every part of the brain, like all the neurotransmitters which are coming out will be imbalanced. Like the stress hormones will be more acting on the brain. And also Alzheimer's disease and Parkinson's disease or even epilepsy, bipolar disorder, all these age-related diseases will be easily crept onto us. And also by the everything we need to do some exercise. For doing the exercise, everything we need to have a good time. That this time and everything is also again determined by economic factor and also social factor and psychological factor, because everyone thinks by running every day they can't do all these exercises. So this is an age-old question, age-old problem. This has many rooms of solutions. Now I think we are going in this path so that we can one day we can achieve this immortality, or something like slowing down the aging process or defeating the death. Because once we think about our human civilization, when it was like 20,000 years or 30,000 years, we won't be able to think about like we are traveling in aeroplanes or something, like we have electricity, or we are talking like this, like that. But now it is possible. We can't imagine that we don't have like that, all those things. But when we are going about 10,000 years or something, they may have the human civilization can achieve all these things. Thank you. Thank you, sir. Any questions? If any questions are there.
Thank you, Dr. Swati, for the wonderful presentation. A very interesting presentation it was.
Thank you,
I request Sabita ma'am that we will have your presentation after Akif sir, because Akif sir is having some pre-PhD class in his department. So I request Aruna ma'am to introduce our next speaker, Dr. Akif Mohammed. Aruna ma'am, over to you. Aruna ma'am, where is she?
Yes, sir. Good afternoon. And it's my pleasure to introduce today's speaker, Dr. Muhammad Akif, sir, who is serving as Assistant Professor at Department of Biochemistry, School of Life Sciences, University of Hyderabad. And he has obtained his graduation with subject chemistry from Aligarh Muslim University, Aligarh, and his master's degree, MSc in biotechnology from Aligarh, and obtained his PhD from Center for DNA Fingerprinting and Diagnostics, Hyderabad, in the year 2008. And title of his PhD work or PhD thesis is Structural and Functional Studies of Mycobacterium tuberculosis thioredoxin system. And coming to his work experience or research experience, he served as a postdoctoral research officer at University of Bath, University Kingdom, during 2008 to 2011, and also served as research associate at Center of DNA Fingerprinting and Diagnostics, Hyderabad, during 2007 to 2008. And his areas of research interest are structure and function of macromolecules and X-ray crystallography. And he focuses on protein structure, which provides a basis of understanding the function of proteins and provides a foundation for structure-based drug design. And he used molecular biology and protein chemistry to generate purified proteins and uses the same for the study of three-dimensional architecture at the atomic level using mainly X-ray crystallography. And in addition, he also uses range of various other biophysical methods and his overall aim to understand the relationship between structure and function of proteins and uses information in the development of new therapeutics and vaccines against infectious diseases. And he has published more than 20 papers, and he has attended national and national conferences, and also was person has invited talks. And coming to his honors, he's conferred with many honors and awards, and few of which are his recipient of Raman Fellowship, Visiting Scientist to Cornell University, USA, and awarding agency was UGC during 2014 to 15, and he's recipient of Young Scientist Travel Fellowship Award by International Conference on Structural Genomics during 2006, and he has received SRF and JRF from PSIR and ICMR. And also he received Department of Biotechnology Scholarship from Government of India during 1998 to 2008. And other than this, he's also into academic activities, and few of his achievements are: he's member of TB Structural Genomics Consortium, USA. He's member of Indian Crystallographic Association, Bangalore, and he's member of Indian Science Congress Association, Kolkata, and is editorial board member of Scientific Report, Nature Publishing Group. So with this brief introduction, I welcome and invite Dr. Muhammad Akif sir to deliver his presentation. And the topic of his talk is structure-based identification of natural compounds inhibitor against Mycobacterium tuberculosis thioredoxin reductase, insights from molecular docking and dynamics simulation. Thank you. And over to you, sir.
Thank you very much, madam. Thank you very much. You have given a very extensive introduction that was not at all required.
Thank you, sir.
I'm really thankful to the organizer for giving me the opportunity to discuss of this work of my lab, which we have published recently. But we are going to present on the structure-based drug design. Before going to the exact result, what we have got it, let me explain what is the structure-based drug design, because I hope the audience are some of the students, and then it would be quite helpful if I can give touch upon some basic. I think there is some disturbance backside. If you can mute.
Abdul Bari, please mute yourself. Mr. Abdul Bari, please mute yourself. Mr. Abdul Bari, please mute yourself.
Sorry for this. I think we need to strictly. Yes, now I think he has muted his speaker. Now, let me explain what is the structure-based drug design. In a structure-based drug design, what we do is we optimize the chemical structure with the goal of identifying a compound suitable for clinical testing. That is called drug. And what is basically based on the knowledge of drugs, three-dimensional structure of biological targets. And what happens that shape and charge of that cause it to interact with the biological target, ultimately that has a therapeutic effect. What is the workflow for structure-based drug design? The workflow basically lies in the three more major steps. One, in case first step is always extraction and purification of target protein. If you are working on the protein, you just either you clone, express in a recombinantly, and then purify the recombinant protein. And those purified recombinant protein you subject for structure determination. There are majorly the three major techniques for the, or in fact, three major techniques for a structure determination is X-ray crystallography, NMR spectroscopy, and very recently the cryo-electron microscopy, which is a very leading technique for structure determination. For that, the two scientists who got a Nobel Prize in 2018 or 2016, basically for cryo-electron microscopy. And ultimately, the third one is homology modeling with the computational way to model the structure. But if you, for the interest of students, those who are there as an audience, I can explain a little bit of what is the X-ray crystallography. Basically, what we purify a protein and then develop a crystal or generate a crystal. And after that the crystal is generated, we expose the crystal on the X-ray and collect the diffraction data. On those diffraction data, which is in the form of intensities, are converted to electron density. Once the electron density is visualized under the visualization platform, what we model atomic atoms in that, and then finally we get the structure. The structure, whenever we talk about the structure, means a spatial arrangement of atom in three dimensions. Atoms could be, when we talk about the structure of protein, means the protein atoms, like amino acids, the atoms in amino acids are arranged in a three dimension, is basically is a structure, and the structure dictates the function. If you know the structure, you can able to use a plenty of information. You get the plenty of information, like you can understand the function, you can understand you can design a inhibitor, you can understand the structure basis of catalysis, all those things. And my lab basically uses X-ray crystallography as a tool to solve the structures many important proteins. I will be giving a few basic information, a few results what we have published recently. But for the interest of student audience, NMR spectroscopy is also a technique where we can get the final user test tool to determine the structure. And cryo is, of course, is there. Now, once you have this structure available, what is the purpose of it? The purpose of, in case of for the theme of this my talk, that this structure can be used for use as a structure-based drug design. Now, what is the purpose? That you identify the druggable protein and the binding site. The druggable binding point cavity can be identified if you have a structure. You use the database from screen the many active compound from the inhibitor database and try to bind it or dock it. And then once you finalize by some calculation, computational calculation, you can always synthesize those compound, and those compound are really tested on the particular enzyme, and finally that can be tested on the whole cell, and that lead molecule can be used as a initial compound for drug discovery. But this is apart from this, you can always use the fragment based approach. Fragment based approach is what happens: you take the smaller fragment of any inhibitor, uh, uh, allow it to co-crystallize along with the protein, and then solve the structure using the X-ray crystallography or any of these techniques what I mentioned in my previous slide, and then identify the position of or binding pattern of those lead molecule or fragment onto the protein structure, and then see the interaction and try to modify those lead molecule further such a way that you can generate a bigger molecule which can specifically bind to your target. So these are the three steps for a structure based or you can say fragment based approach for inhibitor design. Now, my lab using this approach, what we have taken, we use 3 model organisms where my lab focused on is Mycobacterium tuberculosis. Why Mycobacterium tuberculosis? Because I have been working on Mycobacterium tuberculosis from my PhD onwards. Then still I am using that system to work on. And then lately we started some project on Helicobacter pylori, and then very recently I started some project on best structure based vaccine development on Leptospira. But since today I'm going to give some results which we have published recently from the Mycobacterium tuberculosis. Now, let me briefly explain about the Mycobacterium tuberculosis. As everyone knows, the Mycobacterium tuberculosis is the causative agent of tuberculosis, and is a really major health problem in the developing countries. If you find it, tuberculosis everywhere. Those are especially the developing countries. And then if you see the symptoms, is basically the Mycobacterium tuberculosis affect the lung, and the lung is major organ which is being infected by Mycobacterium tuberculosis, and then the infection transmitted from one person to another person. Everyone knows about the Mycobacterium tuberculosis. If you see that if somebody has Mycobacterium tuberculosis, you can always go and get a drug. The medicine is for this one is like though there's a medicine available, but its timing for treatment is 6 to 9 months. Will not go to the detail of that one, but let me explain other thing, like what is the estimated burden of TB burden by if you take the WHO report by 2018 report, the TB incidence along with the HIV, because TB is whenever you have a TB, that there are chances of HIV infection is very common. The TB and HIV is always co-infection person. 2.6 million people are already having incidence of TB cases, and then only TB HIV like if you say in incidence of only TB 92,000, and if we have the other incident of MDR is multiple drug resistance. Same by Mycobacterium tuberculosis, they can develop a resistance against the known and first line of drug, and those cases are also there. If you see my slide, there's the report from WHO, and on the right side of the panel, which shows the cases in India is a district level. We have picked up from one of this news article. Now, very recently, there was a report which says that even the COVID-19 pandemic affects the TB, because the moment you have a COVID-19 pandemic, there are cases that report says that the actual treatment of TB patient has quietly affected, and they have given the projection that additional TB cases can be jumped up by 2020-2025 by 6.3 million. And if you say additional TB death can also be increased from 2020 to 2025 by 1.4 million. Now, with this data, we can understand how significance of really to have some sort of treatment or some sort of management for Mycobacterium tuberculosis infection. Now, what are the challenges? The challenge for this organism is that it's a very slow growing organism. It takes 24 hours to replicate. Hence, the system which we work on this Mycobacterium tuberculosis is quite challenging, because having low, slow growth rate. And it's in the same time, it says that you have a since it's an airborne pathogen, we need to have a BSL-3 labs available where you can handle MTB. And then another thing is called vaccine nature of cell wall. Cell wall is quite vaccine nature, then which prevents the many molecule to enter into the bacteria. Hence, the antibiotic other thing is, or any drug you develop, is quite difficult to enter into the vaccine nature of. And then the most important challenge for this MTB tuberculosis, or we can say microbial tuberculosis, is development of MDR resistant or XDR resistant strain. We may say MDR means multiple drug resistance, or XDR extremely drug resistant strains available, or these which can be difficult to take. Apart from that, the other which I haven't mentioned it is dormancy. Dormancy means the bacteria, even if you have an active TB patient, and if you the patient has undergone for 6 to 9 months of treatment, there are chances that the bacteria, some of the persister, or some of the bacteria which are really resistant to the drug, they can go and sit in the lung, in the glomerulus, and that stage is called dormancy or dormant stage. The dormant bacteria always, once you have active infection, you have always dormant infection present into your body, and it can relapse anytime if your immune system is weak. But this dormancy, the phenomena of dormancy and how the bacteria sitting in the ideal form is still a challenge to develop a drug against dormant bacteria, or develop a vaccine against dormant bacteria. And then, of course, I mentioned MDR and XDR. And then with this challenge is always intend to find a new alternate target. What could be the strategy? In that case, anyone who works on microbial tuberculosis for drug development or vaccine development, they always search for they always design some strategies to cope up with the infection. Like one of their strategy could be either searching and targeting alternate important proteins. Like whatever the targets available, you may always explore the other possible targets which can be targeted by any drug, or targeting any replication machinery, or targeting whole cell bacteria, or multiple targets. Suppose one inhibitor can target multiple pathways. So these are some strategies you keep on following. With this, one of the alternate system which I started my PhD, this thyrodoxine system. The thyrodoxine system is a system of a protein. When I say system, it consists of 3 thyrodoxine, 1 thyrodoxine reductase. This system of protein basically provide a reducing environment into the cell. Now, most of the time, if you consider, like, MTB resides into the macrophages. Macrophages are cells of a human body, immune cells, where the MTB resides. And if you see the nature of macrophages tries to kill the bacteria by providing some sort of oxidative stress, nitric oxide stresses, by free radicals. Those are always is a mechanism of killing of bacteria into the macrophage. However, MTB is so smart enough that it tries to cope up from this hostile environment of macrophages by expressing certain proteins. Among that, one of the protein is the thyrodoxine system, which consists of 1,3-thyrodoxine and 1-thyrodoxine reductases. And with this the function of function was already established some other organism, that thyrodoxine provide a reducing environment. That means if you have a oxidative stress in condition into the cell, these proteins can just minimize those stresses by providing reducing environment, or by preventing from oxidation of the particular protein. MTB, TRX and TRXR, when TRX I say thyrodoxine, and TRXR I say thyrodoxine reductases, involved in reduction of peroxides and nitrobenzenes, and also detoxify the hydroperoxides. And whenever we talk about the target, means the target should be essential. Any gene which is essential, it can be targeted with a drug. With one of the transposon mediated analysis in MTB, it was found that MTB-TRXR, which is thyrodoxine reductase, is essential gene. And since it's a single copy, it makes it quite essential, and hence it could be it can be targeted. And the other information that you need to have with any molecule which is targeted, that it should not have a similar homologous protein in the human. When TRXR was compared with the human counterpart, we found that the human counterpart has with the human counterpart, it has only 23% sequence similarity. That means that it's not quite similar. However, later on, when we were working on that, we found that the structure of TRXR from human is also quite different from TRXR of mycobacterium tuberculosis. TRXR from mammal or human is 55 kilodalton protein, which is a high molecular weight TRXR, and whereas the bacteria, MTB and E. coli, this is called low molecular weight TRXR, which is 35 kilodalton. Apart from these differences, there are the other differences of TRXR. That TRXR in bacteria, the mechanism of catalysis of TRXR is slightly different from, or in fact, different from human counterpart. We have solved this structure in during this time, and we found that using the X-ray crystallographic approach, we found that it has a structure, three-dimensional structure. Overall structure shows it has a two-domain protein. One domain is called NADPS domain, another is FED domain. And then there are two cofactors. One is the FED bound to the FED, and based on this domain is defined based on the binding of this cofactor. FED bound the side, and the NADPH bound to this side, and we have active site which is 216 residues present in cleft between the NADPH and FED domain, pointing toward the isoalexine ring of FED. And this is the overall structure. And what we found by comparing with the other known structures and then conformational flexibility taking into account, when we superimpose the FED domain, what we found, the NADP domain has a quite sort of conformational flexibility or rotation of NADPS domain. So this was published, and then later on, since as a significance, I already mentioned that it can be drug target because it had a single molecule, and there's the similarity with the human counterparts is different. Now, if you see the part of NADPH and FED domain, this two domain is connected by the hinge region, and hinge region hinge region has a two antiparallel beta sheets, and then same time the structure of TRXR along in complex with the thyroxine was published by the other group, and it showed that TRXR in complex of TRX, one of the thyroxine, there's a 66-degree rotation of NADPS domain to provide to bring the active site cysteine from the buried position to the surface, such a way that it can give the reduction potential. The whole mechanism of this one is understood by having a structure of our structure and other structure. That what happens that if you have NADPH pool in the cell, NADPH reduces FAD, and FAD reduces the cysteine of a TRXR, and the cysteine of TRXR, which is buried between the two domain from NADPH domain and FAD domain, and because of change of conformation of NADPH domain, the cysteine residue comes out on the surface and gives the reduction potential from cysteine to the cysteines of thyroxine. And when the thyroxine is reduced, now this thyroxine is not available to reduce any protein which are damaged by the oxidative stress condition in the cell. In our system, in case of mycobacterium tuberculosis. Hence, the mechanism that time mechanism was already understood. Now people have targeted this molecule by different ways. Somebody has targeted this molecule by the interaction of thyroxine and thyroxine reductases. What we was speculating to target this molecule by, since one of the important point which I mentioned here, that the catalysis requires NADPS domain to be rotated by 66 degree, such a way that active site cysteine can come up on the surface to provide the reduction potential to the thyroxine. Hence, we thought, why can we stop this rotation? And to stop this rotation, we have taken this as a target molecule to screen high throughput virtual screening of chemical compound to target this molecule. And this time we decided to target onto the allosteric site or target somewhere which can block this rotation of NADPS domain, and using this, what we have predicted one of the cavity nearby, and that cavity was present between the NADPH and then hinge region and some part of FADH domain. And those hinge region was we have chosen as a cavity where we can screen more than 20,000 compound from eye screen to be docked ahead there. And after docking of this more than 20,000 compound, we found that the two compound 1 and compound 2 shows with very good score of binding on that cavity, and there which is lying between FAD and NADPS domain. And these two compounds, if you see the features of these two compounds, is the molecular weight of this compound was 400 gram per mole, and compound 2 is 328 gram. And then if you see other parameters really required for any drug to have, that it should not be more than 500 gram per mole. That's a Lipinski rule of 5 Lipinski rule, that it should not be more than 500 any drug. And then if you consider other hydrogen bond receptor, etc., how many is that rotational bond toxicity, these are compound were non-toxic. So these two compounds we selected for further their binding study using binding analysis. When we compare this binding analysis, we found that which all atoms are interacting with these two compounds. And what we found, the most of many the residues which are interacting with this compound are mostly the hydrophobic, having hydrophobic interaction, few ionic interactions. And then to check the stability of these two compounds in the protein, we have done the molecular dynamic simulation. As you know, for the audience, student audience, the molecular dynamics simulation is a computer simulation in which the atoms and molecules allowed to interact with each other for a period of time. A molecular system consists of vast number of particles, so it's impossible to find the properties of each complex analytically. So the MD solved the problem by numerical method. The interface between we can say the molecular dynamics is the interface between the experimental and theoretical experiment. And based on that, what we get, these are the protocol we have used, and what we get, we have checked the after the molecular time simulation, we visualize 3 parameters, usually RMSD. RMSD is the root mean square deviation from the previous from the initial structure, and we also check the RMSF, root mean square fluctuation of each atom, and then we check surface solvent accessibility surface area of each compound in complex with our protein target. And what we found, if you see 20 nanoseconds of simulation, these two compounds were quite stable. It doesn't have fall off from I did not get out from the complex. And when we check the RMSF clearly shows some of the region which are quite flexible in the compound into the native protein. However, it's stabilized in the compound 1 and compound 2, which clearly shows that these circle are part of the region where hinge region is present, and these are that clearly show that the binding of this compound stabilize the few of the residues of the hinge region and NDPS domain. And if you see the surface area, some solvent accessibility surface area also is almost similar in both native protein and then compound bond protein. Then we calculate what is the binding energy of the compound. With the binding energy, you can have a binding energy, a van der Waals binding energy, electrostatic interaction, polar solvents, and then surface solvent accessibility surface area, and altogether we have calculated the total also. And when we compare the compound 1 and compound 2, what we found, compound 1 shows is better interaction, having low free energy compared to the compound 1. And most of this this compound is stabilized by hydrophobic interaction. And then same time we have checked the conformational variation of this protein in native protein, or we can say protein itself and the protein in complex with these two inhibitor. What we calculated, which call correlated and anti-correlated motion of each atom if you consider each atom on y-axis and atom on the x-axis, and when we monitor there's a conformational variation, we can monitor it either the atoms are moving in the correlated motion or anti-correlated motion. Those are moving in the blue color, they are all in the correlated motion, and red color is anti-correlated motion. Now, what we found in this case in compound 1 and compound 2, and we have chosen some region, the region of between FAD and FAD, NADPH and FAD region, NADPH hinge region and FAD hinge region, and then we mapped this region onto the correlated anti-correlation motion. But what we found, that's compound 1 and compound 2, the nature of binding of the compound 1 and compound 2 brings about some mixed sort of anti-correlated and correlated motion. And then in region of FAD hinge, mostly strongly anti-correlated motion. And compound 2 is mostly weakly anti-correlated motion in all the region. Now, why we are discussing the conformational variation, that whether the binding of these inhibitors brings about some conformational variation that can be really significant to justify that these conformational variation are really happening in the protein because of inhibitor bound case, will bring about some sort of locking conformation which can block this activity of this protein. Now, apart from this, what we have done some protein contact network. The protein contact network was performed to carefully analyze changes between the interacting residues by comparing the contact map of EPO and HORO complex. EPO means only the TRXR, and TRXR in complex with the compound. The network is a graph where each residue corresponds to a node, and two nodes are connected by an edge. If and only if the two residues are in contact. If the two residues are in contact, we represent as a edge. And the two residues are considered to be connected if they are especially close in the three-dimensional structure and are specified with the two key parameters, the contact type and distance cutoff. And then the contact type defines a subset of atoms in a residue. And the most commonly used cutoff for this, if you consider C alpha based protein contact network, that means you can only C alpha. C alpha means carbon present in the peptide one. Then you have, if you have a cutoff of 0.7 nanometer, we consider that the two atoms are in a contact. Using this is called, in a layman language, you can say how the two atoms are in a close proximity to each other, whether they are forming a sort of nodes or edge that depend upon what their position or distance cutoff we consider it. And then what we found here, when we map this contact protein network contact onto the only TRXR, and TRXR can complex with the compound 1 and compound 2, we can see that how the structural changes are visualized here. That means binding of compound 1 and compound 2 have a different contact protein network contact compared to the wild type or compared to the EPO form of the TRXR. That means that there is some structural changes, or there is some you can also quantify how many interactions are there in case of compound 1 and compound 2. When we quantified, we found that the contacts in compound 1 and compound 2 within the atoms in the protein are more compared to the EPO TRXR. And then if you further, one of the example, if you see the analysis of change between the interacting residues, we have highlighted a few residues where there are plenty of information, there are plenty of contact bonds, but to make it more explainable, we have taken one example. In that one example, if you consider NADPH domain on the top and the below is the FAD domain, and if you calculate the center of mass of these two, the center of mass we calculated here is red color. Here, center of mass of this part we calculated the green color, and then use this center of mass when we visualize or we mapped on the compound 1 and compound 2 bound form. What we found, the center of mass has slightly decreased. Here, the distance is 25 angstrom. However, in these two cases, in this case is 9 angstrom, in this case is 15 angstrom. That means the compound 1 and compound 2 is really brings these two domains close to each other by locking into the one other conformation. such a way that it can be inhibitory for TRX, for NADPS domain to be rotated out, to give the electron, to give the reducing potential, or to expose the cysteine residues on the surface that can pass this reduction potential to thyroxine. And if you further, if you go analyze this structure, what we found that one of the domain, it is in the complex form, compound one. One of the helix from the NADPS domain is having many, or in fact 3 or 4 ionic interaction with the FED domain, and because of interaction, we assume that this binding of inhibitor locking these two domain in one conformation, and once these two conformation are locked by using this inhibitor, by using this compound one, you can also stop catalysis. So this is what assume that whatever the inhibitor we have screened from the database, that can be tested onto the pure protein we purified in the lab. That we are doing it. We are going to procure this inhibitor, and we have already purified our recombinant protein in the lab. We are going to just simply, whatever we have done through the computationally can be tested whether this compound can inhibit this catalysis of this protein. In conclusion, what I can say that the study report the screening of natural compound from one of the database against MTBT-RXR. This led to the identification of 200 compounds, and amongst top two compound 1 and compound 2 were selected, and both compound bound to the hydrophobic group of TRXR, and the binding was stable, which we have checked through the molecular dynamic simulation and the calculation of binding energies. Data obtained in this study conclude that the both natural compound may have a potential to bind. And if we compare from compound 1 and compound 2, I would say that compound 1 is better than compound 2, and this compound can be used as a lead molecule for the inhibition of MTBT-RXR, and this will further validate using in vitro inhibition assay with the purified protein. So this is the conclusion of my part. And then if any work is useless without acknowledging the people who are working on that, if all this work which I present, I would like to acknowledge my student. There are 4 students working in my lab, Pankaj, Nachiket, and Mansiraj, and most of the work which I present here is done by Surbi Lata. She has done all this work. And then I would like to to acknowledge my my collaborator, of course, one of my mentors, Shekhar Shimande, who is a DGCSR. He is my mentor in my PhD. And of course, I have been collaborating and have been using his facility whenever I require. And I'd like to acknowledge my collaborator, Syed Faisal from NIAB Hyderabad. And of course, I would like to acknowledge one of my collaborator from Cornell UC. With that, we have published only one paper, which I haven't presented here, but still is our active collaborator. And then, of course, for funding, the DBT and SERB and UP2 is always acknowledged for this one. And finally, thank you for giving your time. Thank you for listening my talk. If you have any question, I'm really happy to accept that.
Thank you very much, sir. Big thank you, because we are running short of time. If any question is approaching us, we'll be sending you to a mail. Sir, thank you very much for accepting our invitation in a short time. And department, on behalf of Department of Botany, Telangana University, I express deep sense of gratitude. Now, our next speaker, because of technical difficulty, she couldn't present. I request Dr. Sabita ma'am to start her presentation. Over to Sabita ma'am. Thank you.
Thank you, Dr. Ahmed. Thank you.
Dr. Pawan, please bear with us because we are running delay. Dr. Pawan, please, soon after Sabita ma'am's talk, you will be the person to talk. Dr. Pawan, are you there? Okay, Sabita ma'am, you can start sharing your screen, ma'am. Sabita ma'am.
Yeah, yes. Can you hear me, sir?
Yeah, you are audible.
Okay. Can the screen is visible, sir?
No, your screen is not visible.
Hello. क्या अरे समझ में आया
Hello.
Ma'am, you can share screen. Your screen is still not visible.
अच्छा वो स्ट्रक्चर पार्ट तो बहुत डिफिकल्ट होगा उसको शॉर्ट करने के लिए 30 मिनट से ज़्यादा हो गया रूम तो शेयर स्टॉप कर दिया शेयरिंग हाँ स्टॉप ले स्टिल अगर गिर गया दोबारा करता हूँ अरे हैंग हो गया मतलब हो गया ना हाँ सिस्टम हैंग हो गया मेरे साइड से तब सब अटेंड कर रहे थे
Ma'am, your screen is visible now. But sound, ma'am, sound is not available. Unmute yourself, ma'am.
Okay, now is it okay,
Yeah.
sir?
Yeah, okay, now okay.
I have, sorry, extremely sorry for the disturbance. So straight away I'll go to my topic. Actually, the topic of today's is that in vitro production of secondary metabolites from the tissue culture. So why we actually are going for the plant tissue culture? This is an important tool of biotechnology which can help in the conservation of important plant species as also large scale multiplication for the commercial production and in vitro production of the secondary metabolites. So I have just explained that why really we have to go for the in vitro. In vitro means in the tissue culture. Why? Because the plants are already producing many secondary metabolites. Why? Means because the most of the chemicals which are present in plant are very low in quantity. So sometimes it's very difficult for them to isolate and purify these complex structures from the plants. And also there is a lot of variation in the seasonal as well as the geographical variations in the plants regarding to the secondary metabolite quality as well as the quantity. So in this scenario, we have to use then develop an alternative technology, and that is where the tissue culture makes an important role, and it also helps in the conservation as well as the saving from the extinction and endangerment of many of the tissues. So there are various strategies are used for the production of secondary metabolites. We need to manipulate the things so that you can get the maximum production of the secondary metabolites. So one of the important criteria strategy used is the optimization of media for the conditions. So the tissue culture, most of there are different types of media are used. So every part and parcel of media, that is the chemical constant as well as the carbon source and other plays an important role in the quantity of the secondary metabolites. For example, the constants of the media, the carbon source, vitamins, phytohormones, oxins and cytokines, whatever we are using, they play a major role in the production of a metabolite. So we have to manipulate, we have to standardize for each plant to get the maximum secondary metabolite productions. Not only the culture medium, but also the culture conditions in which we are growing, like the temperature, pH and the light, which we are using for the growing of cultures, also plays a major role in production of metabolites. So we have to manipulate these things to get the maximum production. Not only this, the nature of the cultures. So what does it mean by the nature of the cultures? So there are different types of plating of the cultures. There's high density cultures and low density cultures, as well as also to select the important cell lines to get the maximum production of metabolites. So these are some of the conditions which we need has to manipulate and standardize for the production of the maximum secondary metabolites. For example, the media, if we take it, there are different types of media. So there are many studies which has been done in the various medicinal plants to know that which media will give the maximum extent. So that all depends on the plant to plant. Some plants, they have the specific media, and some I will just a few examples I can give. Zheng et al. In 1978 he has studied that the production of serpentine from which is a form of indole alkaloids, and they found that Murashige and MS media, that what we regularly use media, Murashige and Skoog media produce more suitable for production of alkalides in Catharanthus roseus cell. So they could get approximately 2 mg per liter. Catharanthus roseus and the serpentine other alkalides are very, very low in intact plant. They are 0.005% are present in the intact plant. So they can get up to the 2 mg of this by manipulating the culture conditions, especially the media. So this is the one example which shows that the medium plays an important role in the production or the quantity of the metabolites from the medicinal plants. And the next important criteria is the carbon source. We know that mainly sucrose and glucose are the major carbon sources which we use in the tissue culture. So the carbon source affect the cell growth as well as the yield of secondary metabolites in many cases. For example, there is one what we have the rosmarinic acid, which is an important secondary metabolite isolated from the Salvia officinalis cultures, and they could get through 3.5 grams per liter of metabolite, that is the rosmarinic acid, and they use the 5% of sucrose. And if you use the sucrose, if you lower down the concentration of the sucrose to the 3%, the metabolite production has come down to 0.7%. So this clearly indicates that if you standardize the carbon source, you could get the more amount of the secondary metabolites for the production through the in vitro cultures. So that means we need to standardize the culture conditions as well as the media, carbon source, and also the phytohormones. So various types of phytohormones we use in tissue culture to produce the callus as well as for the organogenesis from the suspension as well as the callus cultures. So mostly the auxins and cytokinins which we are used in the kinetin like cytokinins like kinetins influences the growth as well as the productivity of the metabolites. Most of the secondary metabolite productions, the auxins are used at the low concentrations. Why? Because the auxins, they promotes the callus production. That is nothing but the de-differentiation of the cells. So what does it mean by de-differentiation? Already we are inoculating with the explant, which is a differentiated plant organ. When you are keeping in the high concentration of auxin, it is de-differentiated into the callus. So there, if you are using the auxins, it results in the more of callus production. So if you are looking for only for the production of secondary metabolites, generally lower concentration of auxins are used. But this is not always true. There are certain plants where even the high concentration of auxins are also induced more number of secondary metabolites. For example, in case of Mucuna purensis, the secondary metabolites which we use is the L-DOPA, which is used in the treatment of Parkinson's disease, and also disogenin, which is a hormone replacement therapy they use, and this is isolated from Dioscorea deltoides, and they found that 2,4-D is more concentration of secondary metabolites are available. So this means that some of the medicinal plants if we are using auxins at low concentrations, they are giving more amount of secondary metabolites. In some of the plants, if you use the less concentration, it is giving more amount of secondary. So that means you have to standardize the phytohormone concentrations for production of secondary metabolites for each plant. There is no uniform criteria. And another important criteria which we use the dense, that is the callus cultures. We already talked that there are high density callus cultures and low density callus cultures. Generally, for the metabolite productions, the high density callus cultures are preferable. For example, in case of Coptis japonica cells, the highest yield of berberin. Berberin is an important medicinal plant isolated from produced from the Coptis japonica, and they could produce 3.5 grams per liter when intracellularly, when the callus culture mass is 50 to 55 grams per liter. So that means the cells are in more density, they produce more amount of secondary metabolites. So this is the most of the studies has proven this, and some, of course, there is some exceptions, but mostly for the secondary metabolite, high density cultures are preferred. And other methods which we use the high producing strains. So what is the high producing strains? It is nothing but the cell lines. When we are using culturing, we have to select the strains which are producing high amount of secondary metabolites. Why? If you observe that the physiological characteristics of individual plant cells are not always uniform. Some are actively dividing and some they produces secondary metabolites, and we call it as a producing cell lines, and some they don't produce produce secondary metabolites, we call non-producing cell lines. So we have to identify which are the producing cell lines and subculture them and maintain, and those will only give the metabolites. So non-producing cell lines are not useful for the production of metabolites. For example, the cell lines of Catharanthus roseus, which accumulate a high levels of asmalicin and serpentine when they have identified certain lines which are producing the secondary metabolites. So that means the cell lines which you have subcultured in suspension cultures or callus cultures, you have to select the high yielding cell lines, and that they have to maintain for the production of the metabolites. So these are the three important criteria where we have to take up for the secondary metabolite. One is the manipulation of culture conditions, and the second one is the medium, the selection of best media, and a selection and characterization of the hormonal concentrations required for the large amount of the secondary metabolites. So these are the three criteria which we generally use, strategies used for the production of metabolites, and other criteria which we use immobilization of the plant cells. It is nothing but the plant cells, high yielding cell lines, they are covered with in matrices like agar, agarose, alginate, and some other substances which are suitable for immobilization and matrices. So what is the advantage of this immobilization of these culture cells is they will extend the viability of the cells. So if you have identified the cell lines and if you are immobilized with some other chemicals, the viability of the cell lines is maintained, and it is very much useful for the bioreactor or downstream process, how it can be used for the fungal cultures. So this is the one of the technique which generally used standardized for the production of secondary metabolites. For example, there are certain cases which you have studied that in the Catharanthus roseus, the serpentine, and in the case of Morinda citrifolia, anthraquinones were enhanced when they are immobilized state when compared to the suspended cells. And with also they have found that capsicum prutensis, when immobilized on polyurethane foam, they produces 50% of the capsaicin more when compared to the normal cells. So this is a certain example which will give you like if the cells are immobilized and there is the viability is maintained, it is there steadily releases the metabolites into the medium. So this is also one strategy used for standardized for the production of the metabolites. And the third strategy which is the addition of precursors. So what is the precursors? Or nothing but there are related compounds. Sometimes they stimulate the secondary metabolite productions. So the one of the important criteria what we have to use when you are selecting for the precursors is we have to select the inexpensive precursors, because if you are expensive then it is very difficult to get or minimize the cost in productions. So generally majority of the amino acids or acts as a precursors for the biosynthetic pathway of these secondary metabolites. So generally the amino acids, various types of amino acids, they are inexpensive also. So they are used as a precursors and they are added into the various concentration into the medium in which the cells are growing, and the secondary metabolite productions are increased. So for example, they have found that amino acids have been added to the cell suspension cultures for production of propane alkalides, indole alkalides, and ephedrins. So they could see that more amount of the secondary metabolites. And for example, in case of Salvinia officinalis, phenylalanine is one of the biosynthetic precursor of rosmarinic acid. Rosmarinic acid is an important constituent of Salvia officinalis. So when this phenylalanine is added as a precursor into the suspension cultures, it increases the production of rosmarinic acid. So this, there are some examples where it can show that that addition of precursors. For example, tropic acid to the medium of Scopelia japonica increased the amount of alkaloids up to 14 times. So this is the best strategy used for the enhancement of secondary metabolites. So one is the standardization of the culture conditions, and second one is the addition of precursors, and mobilization of cultures, and fourth strategy used for the elicitors. So what are there are various types of elicitors which elicit or increase the secondary metabolite productions. So there may be endogenous, which are present within the plant cells like pectin, pectic acid, celluloses, etc. There may be exogenous that are can be added into the medium like chitosanes, glucans, and chitin. Some are there biotic elicitors, maybe a bacteria, fungi, and so on, and abiotic elicitors like the chemical and other physical factors. So these are the different types of elicitors are used in vitro conditions to enhance the secondary metabolite productions. For example, the chemical elicitors, generally they are used as heavy metals, nickel, cadmium, etc. And the chemical hormonal elicitors like salicylic acid, jasmonic acid, methyl jasmonate. So among all the elicitors, these are the salicylic acid, jasmonic acid, and methyl jasmonate. These are used as to increase the secondary metabolites as an elicitors, and physical factors like light, thermal stress, salt stress, osmotic stress, dot stress. These are also add. So we know that majority of the secondary metabolites are as a product of in the defensive in nature. So they are produced in the plant as a part of defensive mechanism. So when the stress is induced, either a biotic or abiotic stress is induced, it activates the metabolism of secondary metabolites, and that could be the reason when you use the chemical and physical elicitors, the more amount of the secondary metabolites are produced. So these are the chemical and physical elicitors used, and the other the biotic elicitors like fungal species, claviceps, fusarium, Aspergillus niger. So these are the different types of fungal which causes a biotic stress in the Calis cultures, and that increases the secondary metabolite productions. And the similar way, the bacterial like Bacillus, Staphylococcus, Pseudomonas is generally used to induce the secondary metabolite production. Not only that, the compounds isolated from these organisms like pectin, chitin, chitosanes, yeast extract are used for production of the metabolites. So these are the some of the elicitor mechanisms which are used in vitro cultures to enhance the secondary metabolites. For example, the cell lines of Papaver somniferum accumulated sanguinidin when exposed to the homogenate of the fungus Botrytis. So they could get more amount of the metabolite. The similar way, the micellar extract of the Rhizoctina solani fungus increases the production of sesquiterpene in Hyoscyamus muticus. So these are the some example, but there are vast number of examples where you can see that addition sorelin production in parsley, diazosin production in Mexican arm, and there are some other where the yeast extracted used for production of the various secondary metabolites from different species of the plants. So that means the elicitor, any other form, either maybe the physical or maybe the chemical or maybe the biotic, they're used to induce the secondary metabolite production. That way, the compounds are produced in excess when compared to the intact plants. So this is the one. And the fifth strategy which we use is the biotransformation. We know that biotransformation is the mechanism where the suitable substrate or compound may be transformed to the desired product using the biological organisms, maybe the plant cells, the fungi or bacterial enzymes, and so on. So this is very, very important when we are conversion of less important compounds to the valuable metabolite. So this is a new area which is gaining importance in the secondary metabolite. Sometimes is compounds which are of less commercial value is used to get the product which is having the high commercial value. So this is the important for the biotransformation. It is very similar to the analogous to the what we see using the microorganisms or fermentation technology. So you can just use the substrate and use the different organisms, and you can get the product. Sometimes the advantage of biotransformation is sometimes you get the completely a new compound which is not there in the particular plant. So this is the one area which is catching up in the production of secondary metabolites. For example, biotransformation of beta-methyl digitoxin to methyl digoxin. So using digitalis lanterna cells. So if you see that digoxin has a highest large market value, which as an cardiac glycogen. So this is very, very important.
No, sir, see, half an hour we can wait. It's nearly about 1 hour.
High compound of commercial important. In fact,
I
this
have some other meeting.
Company,
I will message him.
they are producing by digoxin by this. Similarly, there are some other organisms, plants like anti-tumor drug, vincristine and vimbrostin has been isolated from Catharsis rogens by using the biotransformation technology, and this was commercialized by Mitsubishi Petrochemicals Limited in Japan. So these are some of the areas where the biotransformation technology is using for the production of the secondary metabolite production. So these are some of the strategies which, and the most important one which now taking up along with the biotransformation technology is the hairy root productions. So hairy root production is the one which is used for the production of secondary metabolites from the origin of the roots. If the roots are having the bioactive constant, you have to completely uproot the plant. So instead of uprooting plants, if you develop the hairy roots, you will get the same type of chemical compounds from the hairy roots. In fact, the hairy roots are produced by the genetic transformation or using Agrobacterium rhizogenesis. And this Agrobacterium rhizogenesis is a natural pathogen which infects the plants. So it integrates its genetic material into the host and it induces production of hairy roots. So these are the hairy roots which you will get in the life. So the important character of hairy roots are they are very fast to grow, they can go very fast, and they are negatively geotrophic. Normal roots are positively geotrophic. These are the negatively geotrophic, and one important character is they can grow on hormone-free medium. Once they are induced, you can just cut them, subculture into the hormone-free medium for many generations, like how you can use for the bacterial cultures, how you can use the fungal cultures. You can use this hairy roots continuously subculturing, and you can harvest the metabolites from the hairy roots. This is a suitability for fermenter system, and this is also being scaled up for the bioreactors, and this is of having the long just industrial applications. So this is the one strategy now they are using for the in vitro production of secondary metabolites by hairy roots, especially if the bioactive compound is present in the roots. So this is the one the important medicinal plants from the extinctions. So you can see that there are many examples where it has showed that the more amount of secondary metabolites, hairy root clones of Hyoscyamus plants grow faster than ordinary root, and they produce a high level of tropane alkalides. So they found that the more amount of alkalides are produced. In the similar way, Hamley et al. Have shown that 17-day-old cultures of hairy roots from the beta vulgaris as twice as much as beta cyanin and 3 times as much beta xanthin than the seedling roots. So this indicates the metabolites are produced in a triple or more amount in the hairy root cultures. So hairy root cultures of Datura stromanium produce Hyoscyamus at the mesotropine. So there are many studies now, and in fact, this is the technology now widely used for production of in vitro production of metabolites, because once you standardize the hairy root production, you can just subculture without any hassle because it's go in a hormone-free medium. And another strategy now which is evolving and which is widely accepted is the endophytic fungi as the source of secondary metabolites. So we know that endophytic fungi are the symbiotic organisms which lives in plants without any symptoms. They don't cause any diseases. They are symbiotic along with the medicinal plants. So this was this symbiotic relationship existed for many years. So what happened during the process of this co-evolution, the interaction of metabolic interactions developed and the production of similar compound to the host. So the fungi is also now able to produce the metabolic product similar to the host plant. So this is very, very important criteria now exploited for the production of the secondary metabolites, because the isolation of fungus and purification is fungus is very easy when compared to the maintenance and production of tissues. And for example, and if you have really a tree species are there, the production through tissue culture is very difficult. The old, the tissues are very old. It is very difficult to establish the callus cultures. In certain conditions, the endophytic fungi can be isolated from this important medicinal plant, and they can be used for the production of secondary metabolites. This is an alternative to the plants which we can use. So these are the different strategies now we are using for the production of secondary metabolites. So which can save, conserve the important plants from the depletion from the natural source. So in some of the work what we have done in secondary metabolites, so we have produced the secondary metabolite diazoxanine in the callus cultures of the Cactus pictus, which is commonly known as the insulin plant. So this Cactus pictus we have isolated through the callus cultures, and we can identify, diazoxanine content more in quantity than what we could see in the rhizomes of the plant. So this is the one of the work we have done in the plant through abiotic elicitor salicylic acid we have used in the callus and suspension cultures of the Cactus pictus. And we also done through the callus cultures the production of embelia from the plant Embelia ribis, which belongs to the myrcinaceae. This is an important compound which acts as the anti-tumor and anti-cancer activity. And this is plant is Embelia ribis is now it is only confined to the certain areas of the Kerala, and it is not available throughout the India. So we could get the quantity of embelline twice what we could get in the seeds of Embelia ribis through the callus culture. So this is the work done in our laboratory. And we also done the production of spillanthal from the Spillanthus acmella, which belongs to the family Astraceae. So this is an important antimicrobial and anti-malarial drug, and this is also used in the toothpaste industry as well as the cosmetic industry. So we could produce the spillanthal by using the hairy root cultures. So we have developed the hairy roots from the leaves and other plants, and we could get more amount of the spillanthal through hairy root culture. So these are some of the work we have done in our laboratory for the production of in vitro production of secondary benefits. So this is an alternative mechanism, not only depending on complete plant, you can develop the mechanism, because the way we are using the plant continuously, when we are not using sustainably manner, we are using, because there are a lot of interest in the natural compounds, the plants are devastating, they are destroying the medicinal plants. So this is the one mechanism where we can use an alternative pathway for production of secondary metabolites. And I'll just give you some of the examples. Actually, this is a technology which has played the very important role, and some successful examples like Taxus baccata, the Taxol production. This is the first FDA approved natural drug. So the drug which you use, the Taxol, is 0.017% dry weight. So this is present very, very low in quantity. And now we know that Taxus baccata is now endangered. It is only limited to not more than 16 plants in the Himalayan regions. So this case, now the work is going on in the many of the laboratories around the world for the production of Taxol, and some success has been achieved it, but still not commercialized it. And also they have produced the first commercial scale production using biolipstick. This is the plant which they have Litospermum erythrorhizon. This produces a natural red color dye which is used as a cosmetic and food additive. So we call it as a lipstick because it is the natural colorant. So this is first commercially produced using the tissue culture by Mitsubishi Petrochemical Limited. This is now FDA approved and they are producing it. And the work is also going around the world for the production of the indole alkaloids in Catharanthus and Catharanthus rosis, because there are more than 130 different indole alkaloids are produced, which includes the vinblastine, vincristin, and sonto, which are having very anti-tumor and anti-cancer activity. But if you see, the percentage of these alkaloids are very, very low, 0.0005% for dry weight. So we have to harvest a large amount of material to get the secondary metabolites. So the tissue culture has now emerged as an important alternative. And the work is also going on production of the saffron. You know that this is the world's highest priced spice, which we'll get from the stigmas of the Crocus sativus. It produces two pigments like crocin, saffronol, picocrocin, and we know very well that 50,000 hand-picked blooms are required to produce 1 kg of dry saffron. So that could be the reason for the highest price. So the work is going on, and the little success has been achieved for the production of the saffron from the callus cultures of the tissue, but still it's not commercialized. And this is also one work which has been going interest around the world, production of quinine, anti-malarial drug from the Quinina officinalis. We know that it is produced from the bark of the Quinina officinalis, and the bark will develop only to 7 to 12 years of growth, and it contains very, very low amount of the quinine. So that's the reason, if you're continuously using the bark, then the growth and development of plant will be stopped. So this is also work is going on, production for the quinine through tissue culture as an alternative measure to save the cinchona trees. And also there is the Dioscorea deltoidea, which is used as a natural estrogen therapy. They have the lot of amount of the corticosteroidal drugs, and this is nowadays used in the estrogen therapy, and this is also a lot of commercial importance. They are doing this work. And another one, which I'll just take this example, like which they have in the Mucuna purins, which produces L-DOPA. We know that dihydroxyphenylalanine, which is used in the treatment of Parkinson's disease. This is the only drug approved for the treatment of Parkinson's disease from the plant based. We know that the Parkinson's side, a disable disorder associated with the deficiency of dopamine in the brain. So this L-DOPA will increase the production of dopamine in the brain. So this is a lot of commercial value. Now they could get the callus accumulate. There are certain works which reported that accumulation of 25 mg per liter of L-DOPA in the medium with the 240. So these are some of the success stories regarding to the in vitro production of secondary metabolites. So I just want to that there are the various strategies which are developing through plant tissue culture, the production of secondary metabolites, because if you are continuously exploiting the plants, ultimately we are left with only few plants. So we have to develop various other biotechnological tools for the for the production of secondary metabolites. So this I made it a very short, brief presentation, because most of the time has been behind. So this is in short. So once again, I'll thank organizers for giving me the opportunity. So if you have any doubts, please welcome.
Ma'am, as we are running short of time, and thank you very much on behalf of the Department of Botany for a wonderful and colorful presentation on the complete in vitro propagation and production of thank you very much for sharing your research experience. Now I will like to request Professor B. Vidyardhini Madam to give her remarks on today's meeting happenings. Over to Vidyardhini Madam.
Good afternoon, everybody, and thank you, Haleem Khan sir. So it's indeed a privilege having everyone on the second day of the International Conference on Emerging Trends in Life Sciences, ICELS 2020, organized by the Department of Botany in virtual mode. So the day 2 session started with Dr. Irfan Ahmed sir giving his talk on rice genomics and bioinformatics for disease resistance. And we had Professor Altaf Ahmed sir, who spoke on genetically encoded fresh based nanosensors, a tool for in vitro measurement of metabolites in living cells, and Dr. Ahmed Ali speaking on nutritional and health benefits of spirulina. Then in the technical session 4, we had Dr. Sabita Rani speaking on biotechnological aspects and in vitro production of secondary metabolites for sustainable utilization of medicinal plants. And we had Dr. G. Swati speaking on new dimensions in undertaking aging processes, as well as Dr. Mohammed Kaif speaking on structure-based identification of natural compound inhibitor against M. tuberculosis, thyroxine reductase, insights for molecular docking and dynamic simulation. So the whole of today's session, we had different speakers, eminent speakers speaking on the emerging trends in life sciences, the different latest aspects of life sciences. And even yesterday, we had very good speakers like Dr. Sham Bandari, Dr. Santosh Kumar, Dr. Insaf Ahmed Qureshi, Dr. Biju Dharmapalan, Dr. M. Tippaswamy, and Dr. Harish Handral. So on the whole, whatever we have shown, we have tried to present in this. I hope all the participants have enjoyed, and we thank each and every one for their presentations, the resource persons, and we thank each and every participant for the patient listening. And I hope all of you enjoyed. Whatever you want, you can just write it to us, and we will send back the replied queries. Thanks to everyone for your patient bearing. Thank you and God bless. Over to Haleem Khan sir.
Thank you, ma'am. Now, quick response from Aruna ma'am. Aruna ma'am, over to you.
Hello, sir.
Yes, ma'am. You're audible and visible also.
You are asking me to sum up the program, sir.
Yes, we have to finish off the.
We have got one more lecture, sir.
Pawan sir has a meeting with the Vice Chancellor, so he couldn't. His time was 1:30 to 2 o'clock, and he couldn't join us now.
Okay, sir.
Okay.
Thank you. So good afternoon, all the dignitaries, all the resource persons, and all the participants. On behalf of the Department of Botany, I would like to give a few points which we have learned from this 2-day international conference on emerging trends in life sciences on virtual mode. And as it was day 1 on 15th of December, it was inaugural session, and the gathering was addressed by Dr. Mahda Bamji Madam, where she has focused on all aspects of life sciences, and she has also given some recommendations to be followed in the society. She has spoken about the food security and all about the principles of genetic engineering. And then it was inaugural address where we had our Resta Madam, Professor Naseem Madam. She had given an overview of the conference. We are thankful to you, Madam. And we have divided the conference into sessions. And day 1, we had technical session 1, and where we had a talk by Dr. Shyam Kumar Bandare sir, and he has delivered his extensive lecture on extracellular vesicles. And we are very much thankful to you, sir, where we have learned all about the classification, biogenesis, functions, isolation, and purification of extracellular vesicles. And you have also focused for some images on TEM. And nanorite is a machine which was used to measure the size and number of nanoparticles, and all about the proteins enriched in cell vesicles. So all this was learned from your side, and we have also shared for your experiences in research lab which was going on. And then it was followed by talk by Dr. Santosh Kumar sir, and he talked on medicinal plants and that how safe are raw herbal drugs. And it was really interesting, sir. And you have all told about differentiation between substitution and adulteration, and why there's a lot of confusion among the people using all vernacular names. You have given some examples regarding indigenous system of medicine and local names. And citing some examples, you have made it very clear what substitution and how it was differing from adulteration, and how it was useful to identify the species and trait. And you have used for some of the techniques. And one of the important technique in molecular biology was which you dealt was about DNA barcoding, and we are very much thankful to you, sir. And next, it was talked by Dr. Insaf Ahmed Qureshi, sir, and he talked on development of novel multi-epitope vaccine against SARS-CoV using immunoinformatics approaches. And we are thankful to you, sir. And it was technical session 2, where we had speaker Biju Dharmapalan, and his talk was on molecular biology for understanding complexities of life, and all aspects of diversity of organisms. And he has shown a photograph of mushrooms and gymnosperms like ginkgo biloba, and photograph of peacock sitting on the tree. So it was very well explained about diversity of nature. And his talk clearly made us understand about biomolecules like carbohydrates, lipids, proteins, and all about DNA molecule, and also about techniques like PCR. And next, it was the talk followed by Dr. Harish Handral, and his talk also was somewhere correlated to the previous speaker. And he was talking all about pluripotent stem cells and types of pluripotent stem cells. And he had given a comparative account of roadmap of drug discovery. And he also talked about 3D bioprinting and organ bioprinting. It was really helpful, and hope that all our participants have learned from your deliberation. And thank you, sir. And next, it was talked by Dr. M. Tippeswamy, and he talked about RT-PCR and its application in crop plants. And it was wonderful, sir. We are very much thankful to you. And it was day 2 today, and we had technical session 3 and 4. Though we had some disturbances and hurdles, we had completely and successfully completed the program, and we are thankful to all the resource persons of today. And it was Dr. Irfan Ahmed Ghazi who delivered his talk on rice genomics and bioinformatics for disease resistance. And it was Dr. Ahmed Ali who spoke on nutritional and health benefits of spirulina. And we are very much thankful to you, sir, as I fall from the same category like algology, where I have done my research in Cladophora. I was very much interested to hear to your topic on spirulina, and it's best food for tomorrow as declared by WHO. It is a superfood, and you have covered all the aspects of therapeutics and all properties of nutraceutical benefits. And it's that alga which is a source of food, and people are not aware of having this spirulina capsule where it enhances the immunity or boosts the immunity. And we all depend only on fruits and vegetables. So we living in society should also know something about this alga, which can be used as food, which can boost our immunity, especially and exclusively during this period of pandemics. Thank you, sir. And next, it was talk by Altaf Ahmed, Professor Altaf Ahmed, sir, where his talk was interesting. And next session, it was Dr. G. Swati. She had made the talk very interesting and lively, and we are thankful to you, madam. And earlier talk was about how to enhance the lifespan in human beings, and how the telomerase is functional, and how it plays an important role in increasing the human lifespan. It was really interesting, and thank you, madam. And it was also talked by Dr. Muhammad Akif, sir, where he had shared his experiences of his research. Thank you, sir. And it was Dr. Sabita Rani, madam, who is our friend and who's very much attached to us. And she is very much regularly into work of our university. And thank you for accepting our invitation, madam. And thank you for delivering your lecture, which was very much interesting. And you have taken almost all the medicinal plants And you have given a very good lecture, which is really helpful to all the researchers doing research in that area. And with this concluding remarks, I'm thankful to Dr. Haleem Khan sir for giving me this opportunity. And we also thank our registrar madam, Professor Nasim madam, supporting the department to organize the seminars in a continuous manner. And this is a third program which we are organizing, and that too on virtual mode. And with these few concluding remarks, I'm really grateful to all the colleagues of our department, where we are united, and we will be united. And thank you. And at last and not least, I also thank all the participants and audience who were with us for 2 days. And thank you all for your patient listening, and hope that in your future we'll gather in many more programs. And I thank all the resource persons, and I thank each and everybody who had made this event successful. Thank you, sir. Thank you.
Thank you, ma'am. Jai Hind. So hereby we declare that the conference comes to an end. Dear participants, we will be communicating your certificates by weekend, because we are manually preparing the certificates. So please bear with us. We have communicated you both days feedback forms. Please clearly fill. Once again, I request that please fill the email IDs clearly, and soon we'll be sending you the certificates. Thank you all the speakers and entire cast and crew of the Botany Department, Telangana University, press of Nizamabad local dailies, and our PRO, Dr. Triveni, and his team, Mr. Naresh, and Mr. Naveen, who is a computer programmer who looks the publicity on the website of Telangana University. So once again, I express my deep sense of gratitude towards each and everybody. Thank you. We'll end the meeting over here. Thank you so much.